All right. Well, we are just about at the hour. So I think we'll get started with just some of our introductory material. Um, Thank you, everybody. You are attending. Can everybody hear me, first of all? Can someone give me a thumbs up? Okay, great. Um, Thank you. Um, we are about to have our first virtual applied crystallization workshop. And so for the next several hours, we're going to be talking about how we do things at the National Crystallization Center, tips and techniques that we have about crystallization and general structural biology. We are recording this workshop, and so we will be posting this online in the next bit of time. And so if you have to miss part of it but want to come back and look at it, it will be available. And we will just start by diving in. So I'll start. I'm going to minimize that. Okay, so the first thing we're going to do right now is just a really brief introduction to the workshop and logistics. Daria from SSRL is going to do an introduction to structural biology after that. And then we'll talk a little bit about making macromolecular crystals, finally then using the National Crystallization Center. And then we're going to have a tutorial on using our Marco Polo GUI, including a breakout room if anybody needs help. We'll have a lunch or evening or coffee break at around 1 p.m. Eastern. And then we'll do optimization harvesting. And then we will have presentations from various synchrotrons, SSRL, CHESS, and NSLS2. And we'll wrap up with some final Q&As. I will say, please, if you have questions, go ahead and write them into the chat. We will get those. I'll cover this in a second. Sorry. So this is Buffalo, New York. We are in the National Crystallization Center located in Buffalo, New York. Here's actually the building, probably the office I'm in right here. And the people who are on the call who are going to be, you're going to be hearing from include the team in the Crystallization Center and Christopher Campomizzi. And so you will be hearing from us. We also have a number of speakers who are going to be in the morning and the afternoon session. And so I want to give just a big thanks to all of you who are attending and speaking. So first of all, we've got representation from SSRL, from the Structural Macromolecular Biology Division in California. And so that's Ina Cohen, Dario Marshani Rivera, and Sylvia Russi. Thank you very much for joining us in our workshop. From Brookhaven National Laboratory, NSLS2, Vivian Stojanov will be talking. And from CHESS at Cornell High Energy Synchrotron Source, Jenny Wireman will be telling you about how to send samples to CHESS. So thank you to all of our speakers for spending time and making kind of the effort to attend this workshop. We're really appreciative. We are also extremely appreciative for our workshop sponsors. So this workshop is funded by our national resource funding from NIH and IGMS. And so thank you to NIH for helping to support the crystallization community by supporting the Crystallization Center. And thank you especially to our vendors, Hampton Research, Mitogen, Formulatrix, and SPT LabTech, who have all helped to fund and support this workshop. We're really appreciative for kind of all of the support and all of the great work that you all do. Okay, some logistics. I got ahead of myself before. So Zoom etiquette, first of all, sorry for the typo in etiquette. Everybody, I know at this point is really familiar with using Zoom, but just as a reminder, you will be muted when you come into the meeting, but if you unmute for any reason, please remember to go ahead and mute yourself again. We don't necessarily need to hear other conversations that might be happening. If you have questions throughout the meeting, please ask the questions in the chat. We will be monitoring the chat and trying to address the questions as they come up, perhaps at the intersections between the different sections. And then, of course, you can unmute if you want to ask your question yourself. And so we'll definitely be monitoring and calling people out. If you prefer to have us ask the question for you, to type it in and we'll go ahead and do so. Okay, so we are actually already ahead of schedule. This is great because we are ready to dive into the introduction to structural biology. I'm going to stop sharing my screen to let Daria share her screen. So really, again, grateful, Daria, for you coming and presenting this this talk on just an introduction to structural biology. Daria Marchani Rivera is in the SSRL SMB group, and she is a wonderful beamline scientist. It's great to work with her. And so I'm going to transfer it over to you, Daria.
Thank you, Tara. Morning, afternoon, I don't know where you are exactly. As Sarah said, I will give you a small introduction about what structural biology is. We're gonna go over, okay, why is it not moving? I'm sorry, it's not responding. Okay, about what structural biology is and what we wanna address in structural biology, and some of the techniques that we can use, and talk a little bit about the strengths and limitation. The techniques are aimed to study structural aspects, biophysicals, biochemical, and computational aspects in both all of these. By no means I know all of them by heart. I've done some of them, so bear with me. So what's structural biology? Structural biology is just studying macromolecular, the structure of macromolecules to understand their structure and dynamics and their biological function and understand them. It incorporates principles from molecular biology, biochemistry, and biophysics. And what we want to do is understand the structure of a macromolecule, and to do that we need to understand the primary structure, secondary, tertiary, and quaternary structures, the structural domains and structural motives that the protein has, which are directly involved with the function of the proteins, how the proteins fall to actually be able to carry their function, the shape of the protein to understand interactions with other macromolecules, and to understand the dynamics of the protein, to the point that we can solve problems by identifying drug targets, studying the interactions between the pathogens and hosts, understand the aggregation of proteins that cause health problems and diseases, understand the structure of viruses and other pathogens, and making molecular models of these macromolecules with the aim to create a solve or prevent, treat, and mediate illnesses, and also to make a more sustainable world. This is an example of what structural biology is able to accomplish. This is the nuclear pore complex. Is a molecular machinery in charge of transporting macromolecules like messenger RNA and proteins from and outside the nucleus. This was an effort of multiple techniques available in structural biology to be able to create this model of this complex. It involved cryo-EM and computational methods. So a little into the techniques available out there. This is information obtained from the internet. There's some communities I think that there are more main techniques about this, but mainly X-ray crystallography is considered the most accurate to understand the position of atoms in space and solve the structure through high resolution. Nuclear magnetic resonance spectroscopy is one of the oldest techniques used in structural biology. And more recently, cryo-EM electron microscopy has become a main technique because of the capabilities that it offers for hard to study macromoleculars. Also, to supplement or complement these techniques, there's the techniques of X-ray absorption and emission spectroscopy, cross-linking mass spectrometry, small angle scattering, X-ray scattering, neutron diffraction, proteolysis, circular dichronism, EPR, and other computational methods. I'm gonna go over some of these in more detail than others, but the main thing is that all of them have strengths and limitations, and actually a combination of series of them will give you a better understanding of your protein or macromolecule. To start, how do you choose which technique to use? Each of them, the first limitation is the size of what you want to study. So in this graph, you can see that depending on the size of your subject of study, you will aim to use a different technique. And again, all of them have strengths and limitations to it. We'll start with X-ray crystallography. Like I said, this is one of the most accurate because of the atomic resolution you can obtain from this technique. It involves the use of crystals. We will give you a diffraction pattern, and using mathematical equations, you can devise an electron density map to model and determine the structure of your macromolecule. At this time and point in time, there's a high degree of automation to collect data. So it's fast data acquisition and processing, and you can solve your structure in a day. The limitations of this technique lie in that if you have a large complex, it's difficult to crystallize it. Sometimes, even when you obtain a crystal, it is hard or difficult to achieve high resolution. And it has been shown that sometimes proteins being in the crystal form affect the global molecular structure, which can be relevant to the function. More recently, an expansion to X-ray crystallography was achievable thanks to the use of higher intensity X-ray sources like the X-cells. These allow to do crystallographic studies to determine structure with minimal radiation damage, which can be a limitation in conventional X-ray crystallography. You can use smaller crystals than in traditional X-ray crystallography, and it enables the study of dynamics in the femtosecond time range. In the limitations, since it's relatively new still in science, there's no standard setup and there's not a lot of automation in it. So it requires a lot of effort from multiple groups. It's less accessible than traditional X-ray crystallography. So you have more competition to actually get time to use an X-ray, and it requires a lot of sample. But in the good side of it, you get to have more accurate results about your structural data. A way to overcome some of these limitations on the X-ray is using fixed target setups, which helps in the time frames of getting an experiment done, because it can be more automated. Secondly, we have cryo-EM. Cryo-EM has the strengths that you can use it for large complex proteins. Usually it's useful for membrane proteins which are very hard to crystallize in, and proteins that don't like to actually be in crystal form. They are reaching near atomic resolution, but it's still not there. And the samples are more in solution state than in the crystallized form. In these limitations, cryo-EM can be a very expensive technique. Well, it is an expensive technique. It needs a lot of computational power to go through the data processing, and the sample preparation can be very hard, and it can destroy your sample, the complexes when you're preparing your samples in the grids. And it cannot be used for small proteins on their own because you won't be able to see them. NMR, nuclear magnetic resonance, is, like I mentioned before, is one of the oldest techniques used for structural studies. It's especially useful for biomolecules under near native conditions because you don't have to, you're not changing the protein from their natural environment. You can use it in their natural environment or close to the natural environment. It's very versatile and it doesn't destroy your sample. You can use it to elucidate structural and molecular dynamic features, and you don't need a lot of sample for it. In the limitations, the equipment requires a lot of maintenance, and it's quite expensive. And it has low sensitivity, and it's costly because you need to use isotopes to be able to see the signals that you wanna that you're aiming for. And when you're using larger molecules, it can affect the homogeneity of the sample in aqueous solution. Now we're gonna go to the complementary techniques, which can be very useful. We have X-ray absorption and emission spectroscopy. This technique is mostly used in metal biomolecules. It can help you determine the geometry and electronic structure of your metal centers and identify oxidation states. Small angle X-ray scattering is based on the scattering of X-rays, and it can give you a low resolution molecular model of your protein in solution. You can determine the global shape of it and the flexibility of your protein in solution. And neutron diffraction, which is similar to X-ray, but instead of diffracting X-ray, you're looking into the protons in your protein. And this technique is very accurate also in determining the position of atoms in space, but it's harder to obtain because it requires bigger crystals, very huge crystals. And like you have heard, like I've been saying before, sometimes it's hard to obtain a crystal, and even more bigger crystals like it's needed for neutron diffraction. Electron paramagnetic resonance, EPR, it helps you, allows you to study molecular motion and dynamics, and it can show you the presence of different secondary structure elements. This technique relies on the paramagnetic characteristic of our species. So if your species is paramagnetic, then you can use it. If it's silent, then you won't see any signal using EPR. And lastly, you have computational methods, which has taken a huge boom in the latest years, which can, like AlphaFold, which you can predict the structure of your protein based on machine learning. And then you can use that as a model to solve your structure. Apart from these techniques that you can see mostly the quaternary, tertiary structures, there are other techniques that help you understand the primary and secondary structures of proteins, which include cross-linking mass spectroscopy. It can help you get into information about protein interactions. and it can be very useful on intrinsically disordered proteins, IVs. We have proteolysis, which can help identify the flexible regions and structural domains on your protein. It's a technique that helps you understand the folding of your protein. Circular dichroism also is useful for understanding the folding of your protein. It helps you identify the alpha helices, beta sheets, and random coils you have in your structure. And to finish, I wanted to say that at SLAC National Lab, we're one of the, I think, only labs that we can do a lot of these techniques, especially using X-ray crystallography, cryo-EM, and X-cells. Oops, sorry. Which you can do crystallography, stacks, small angle scattering, SAS, as X-ray absorption, cryo-EM, and the use of the X-cells. So with that, if you have any questions. Can anybody hear me?
It looks like, yep. Looks
I'm
like
sorry.
Claudio, go ahead and unmute.
Hello. In your slides, you mentioned that the NMR is a low sensitivity technique with labeled nitrogen and carbon. Now, since I'm doing that currently, I was wondering, everybody told me that it's a high sensitivity and you can get structure with 1 angstrom resolution. Why do you think it's not sensitive enough? We have an 800 NMR.
It's because you need the isotopes present in your protein in your molecular study. So it will depend on how many isotopes you have there. But if you're I say if you can have a lot of isotopes in your sample, then it will be a high.
What do you mean, how many isotopes? Like you grow bacteria in minimal medium using ammonium chloride and carbon that are labeled as only carbon and nitrogen source. Yeah, like the only carbon source that you use in your, I can't modify. Well, the only carbon source that you use in your culture are carbon-13 and nitrogen-15.
So, Daria, I'm going to jump in because before I became a crystallographer, I was an NMR spectroscopist. So I think that NMR is a really great technique to use for structural work if you can get your bacteria to actually express your protein with carbon-13 labeled glucose or N15 labeled ammonium sulfate. There are definitely some occasions where that actually becomes difficult. And if, for instance, you can't express your sample in bacteria and you have to use different types of expression systems like and different expression cell lines, it can become extremely just a lot more difficult. The other, I would say, limitation in terms of NMR is the size limitation. So I think that with everything, like there's so many different techniques that are available. And the key thing, I think, for all of us is that we want to use the technique that's going to be the best thing for the different sample. And so, yes, if you have a sample that is a relatively small soluble sample that you can label easily, NMR is an absolutely wonderful technique, and I highly encourage you to use it. So.
Yeah, okay. Yeah, it's expressing and yeah, that's thank you.
Wonderful. Okay, so we have a question from Miranda. Can natural abundance isotopes be sufficient for structural work or must you label? And you know, again, I'm happy to answer this. So typically, you know, you don't need to label, for instance, your hydrogens if you're doing just kind of one-dimensional NMR. But in order to be able to actually have NMR active nuclei, you actually need the N15 label and the carbon-13 label to be able to actually do those types of 2-dimensional and 3-dimensional experiments that are pretty critical for getting structural work done. Claudio, if you feel anything differently there, let me know. But because you're probably a more of an NMR expert at this point than I am.
No, I'm not. We are just dealing with a protein which is very small and is not able neither to crystallize nor to get a very high concentration, and therefore the NMR is the only solution. The point is, the natural occurring isotopes, they're simply not enough because you need really to detect the isotopes. It's you can't do without.
Yeah, the natural abundance is much too low to be able to detect for carbon and nitrogen. So
Any other questions?
yep, there's one more question. Let's see. What
And
measures?
thanks-
Huh?
And thanks for jumping in. I'm not an MR
That's okay.
expert at all.
The next one is about cryo-EM. What measures do you take to solve macromolecular complexes less than 80 kilodaltons apart from MAbs in cryo-EM?
That's a very specific question for cryo-EM. So if there's anyone in the audience that knows cryo-EM, I'm a X-ray crystallography, and I don't consider myself a full expert in it, but that's what I do. So if there's someone in the audience that could answer, like, it would be better.
Yeah, and I think that might be a little bit outside of the scope of our crystallization workshop. And let's see, one more question. This is an NMR question again. Would NMR be a good approach for determining ligand protein binding mode in a less than 70 kilodalton? I would say that's probably a little bit large for NMR. It's just at the upper border of NMR. Now, if you have one protein that is smaller and you could titrate in the other protein and actually watch kind of where in the NMR spectra things were changing, that might be a good way to actually map out where the interactions are occurring. So yeah, that's my answer for you, Maying, about that. All right, so to keep on schedule here, I'm gonna have you stop your share, Daria. Thank you so much for that great talk. I love that introduction. And I'm going to share my screen. For those of you who kind of can't tell, give me just a second while I figure this out. I have a bit of a cold, and so I have recorded my talk so that you're not subjected to coughing during the talk. So I just have to figure out how to share it. There we go. And I'm going to make sure as it's going here. Let's see. I'm going to just kind of get this started. Okay, can anybody hear? Can somebody show me if they can hear?
You have to turn the volume up a little,
Okay, let's
but we can hear it. Oh, it might be all the way up. That might be up to the individual to turn their own volume up.
Okay. I do want to just kind of warn you, there will be a couple of jumpy spots in the middle of this recorded video where I stopped because I was having a coughing fit. So apologies for the kind of jumpiness. And we'll do questions at the end of this. So here we go. Okay, so for this session of the workshop, I am pre-recording this, and so please hold on to any questions to the end. I'm have a little cold, and I want to make sure that my voice is available to actually engage with everybody during the workshop. So in this session, we're going to be talking about some of the specifics about different crystal-based methods, as well as what are some of the basic parameter spaces that we need to think about when we're thinking about making macromolecular crystals. And so one of the key things is that all crystal-based structural biology methods really require very similar things. The first thing is, of course, a crystal or a set of crystals. The next thing we need is some type of an energy source. We're going to be talking a lot about X-ray crystallography, which generates X-ray diffraction data and has an X-ray as a source. But there are other methods that can also be used, including X-ray free electron lasers or XFELs, neutron crystallography, and electron diffraction. So the next thing that we need is for our crystals in this energy source to actually diffract. And one of the things we'll talk about later today is what are some of the different techniques that we can use if the crystals are not diffracting well. What are the things we can do to troubleshoot those? And then the thing we're not going to talk about a lot today is solving the structure from the diffraction data. But ultimately, our end goal is to take the different diffraction data sets that we get and be able to put together a three-dimensional structure of our macromolecular target of interest. So I want to highlight here that I think sometimes we think about all of these steps and all of the things that are happening as in some ways a little bit mysterious and magical. And it's true when you think about it. We are taking a very high energy source. We're going to put our crystal into it. We're going to collect a bunch of diffraction data, and then we're going to take a bunch of that diffraction data, put it all together, and solve a three-dimensional structure. And while there are a lot of really magical and wonderful things about being a structural biologist, and this is a representation of what we do, I also really want to get across that a lot of the things that we do and that we're doing to both use different energy sources and create our crystals and collect our diffraction data, that these are all very science-based and that there are physical properties underneath the things that are making all of this stuff work. And so we get to, be a little bit of a magician when we're doing structural biology, but, the science is there. And that's what we're going to talk about. So there are different, like I said, different types of crystal-based structural methods. So neutron crystallography requires these massively large crystals, hundreds to thousands of times larger than what we need for X-ray diffraction. On the other end of the spectrum, we have things like microcrystal electron diffraction. In this case, we need crystals that are hundreds to thousands of times smaller than X-ray diffraction. What that means is that we actually have an extremely large range of crystal sizes that we can make use of. All of these methods actually require crystals, and so I've got just this little time-lapse video of this beautiful crystal growing in. And we, as crystallographers, love to see this type of thing. Just these beautiful crystals growing into a well that would be easily looped up and harvested. And again, we're going to talk about that later in the day too. So I will say that in terms of the different types of methods that are being used, crystal-based methods really account for a huge amount of the structural data that is available in the protein data bank. So as of yesterday, 220,000 structures have been deposited to the PDB, and about 84% of those have been in some way crystal-based. So either synchrotron, XFEL, electron diffraction, neutron diffraction. And so the need to grow crystals and to make crystals is exceedingly large. Crystallization is really the first step in crystal-based structural methods. This is probably a more typical way of looking at that path of going from your sample to screening to find crystallization conditions, to a protein crystal, to your diffraction data, to your phasing and electron density map, until you get to your solved structure. So the first thing you might want to ask at this point is, what exactly is crystallization? So crystallization is the phase separation from a thermodynamically metastable supersaturated solution under the control of kinetic parameters. That's kind of a mouthful. And so we're going to actually now spend the next 20 minutes or so talking about the different parameters that help to encourage our macromolecules to actually generate a crystallized state. So first of all, one of the things we're trying to do is generate a periodic network of relatively sparse and weak intermolecular interactions. This is often driven by the crystallization cocktail and the different components in the cocktail, as well as temperature. And so we really need to vary the parameter space to crystallize your sample, and that is a lot of what we're going to talk about here. Crystallization is really typically a two-step process, sometimes more complicated, but in its most basic state, it's a two-step process. We start with samples in solution, and we ideally end up with crystals that have grown. And so proteins in solution, essentially the solubility of that protein is a function of all manner of different parameters. But ultimately, the crystallization process lowers the free energy of these proteins by a considerable amount. What does that actually mean? It means that your macromolecules really want to crystallize. The free energy, there's actually an energy difference between free molecules in solution and in the crystal form. However, there's also an energy barrier to nucleation. And so we have a period here where we can actually be moving into a space where we're supersaturating the solution just based on concentrating it. And that can go one of two ways. It can go into a precipitation or aggregation, or it can start to nucleate and then generate. So we think a lot about how to describe crystallization by using phase diagrams. And so you are seeing right here a phase diagram that shows essentially a sample in an Eppendorf. It's a single phase sample in solution. On the bottom axis is a precipitant, some type of conditions that are going to encourage that sample to crystallize. The sample concentration is actually on the y-axis, and you can see that there's a variety of different types of crystallization methods that can be used. We're going to talk about some of those. Our goal is to go from being in an undersaturated state into a biomolecule-rich saturated phase that is nucleated and forms crystals before it precipitates and aggregates out of solution. And so this is a very fine balance, and it's a large part of what is sometimes difficult in terms of crystallization. Now, when we think about biomolecular crystallization, probably the thing that you think about the most at the beginning is, what is my crystallization cocktail? What's going to make my sample crystallize? And the thing I want to try to get across is that is absolutely important, and we will be talking about that quite a bit. But the first thing you absolutely have to think about are biochemical considerations for your sample. If you don't have a pure, stable sample in a stable buffer, and you have a good understanding of kind of the concentration and pH and all of these other factors, that sample is probably not going to crystallize. And so we're going to talk about the different biochemical considerations that we have to take into consideration. So there are a huge number of biochemical obstacles in forming protein crystals. And this is actually a slide that I saw a couple of years ago, and I absolutely loved, because I think it really highlights why you have to think about the biochemistry. What you can see in these slides are the different ways that your biochemistry can cause things to go wrong in crystallization. If you have impurities in your sample, you've got different things of different sizes, that's going to mess up forming an ordered lattice. If you have aggregation or precipitation, that's not going to generate a nice ordered lattice of your sample. If you have any flexible domains, like little linkers that are moving your sample around, that is going to really stop crystallization. What if you have a flexible tail on your protein? Gonna make it hard to crystallize. What if you have kind of variously sized proteins because you've got proteolysis? Not gonna crystallize. If you have some unfolding events, not gonna crystallize. If you have pure protein, all of these little paper clips are the same size and, you know, hanging out in this petri dish and very happy, they're going to ultimately form crystals because your sample wants to crystallize. There's a bunch of things that are going to get in the way of it crystallizing, but it's not the protein itself. It's all the different things that are impacting the protein. And so a major key to success in protein crystallization is actually good protein biochemistry. So what are the different things you have to think about in terms of protein biochemistry? You need a very pure sample. And so essentially you really need at least 90%, ideally even a little bit better. There are times when you might actually get crystals to form even in the presence of impurities, but if you do, they're probably not going to diffract very well. So kind of going back to our overall list of things, you need a crystal, and then you need to shoot that crystal with some type of energy source, and then you need to assess the diffraction. First thing, you need to get a pure sample. So run a gel on your sample, overload that gel. You know, it's not the one that's going to go into publication for you, but it's going to tell you that you have a very pure sample. Okay, what are the different sources of impurities and heterogeneity? You know, multiple species, as you saw in the paperclip example, you know, oligomerization, isoforms, a misfolded population, partial proteolysis, different flexible regions, and different kind of impacts on different amino acids. And so, again, a very pure sample. It can sometimes take weeks for nucleation to occur. And what does that mean? It means that your protein is sitting in a crystallization tray in a well, and it has to stay stable without unfolding for that whole time. There are different things that you can do to check your protein stability. So TSF or CD, and these are different methods that can be used to assess the stability of your sample as a function of a lot of different parameter space, like temperature, pH, buffer, salt conditions, having a ligand present or stabilizing chemicals. You also need your sample to be very soluble. So highly soluble, homogeneous sample, there are a lot of different ways that you can think about how to check your sample solubility. The thing that we usually do is dynamic light scattering. It helps us see whether our sample is in fact in a kind of monodisperse, low aggregation state, or whether there's either polydisperse, so that means there's multiple kind of species or some aggregation, or whole all the way up to just completely aggregated. If your sample is aggregated in solution, it is not going to form a crystal. What buffer should your protein be in? So the absolute best buffer is the simplest formulation that kind of keeps your protein happy, makes it soluble, homogeneous, stable, and keeps it active. There are definitely some things that you want to try to avoid if it's at all possible. So phosphate buffers like to form salt crystals. An ideal buffer concentration is under 25 millimolar. Sometimes that's not possible. Salt concentration, we like to try to keep under 200 millimolar. Again, sometimes not possible. And glycerol, of course, is a solubilizing agent, and so it's important to keep your sample soluble, but it will also retard crystallization if you have to use too much glycerol. So our kind of rule of thumb is to try to avoid glycerol if it's possible, and if it's not, to try to make it so that it's around 5% or less in the final crystallization drop. Okay, what about pH? So again, the ideal pH is the one in which your sample is the happiest. And one thing to keep in mind is that the crystallization cocktail is also pH dependent. And so this is a really common parameter that you can modulate during crystallization trials. Those surface charges on your sample, remember back the image of those weak intermolecular interactions, that can really impact crystal packing. And so thinking about pH can be a really helpful thing in terms of how your crystals might be growing. Some samples actually need reductants, and if you need a reductant, you should really think about reductant lifetime as well as the impact of the reductant lifetime as it's impacted by pH. And so different reductants can last for different amounts of time depending. And so sometimes you have to kind of do a little bit of trial and error to see which one you need to use. So next thing, what about the concentration of your sample? So the range of sample concentration that's effective for crystallization is extremely variable. And one of the things you have to kind of remember is that we have to start with a undersaturated sample and make it supersaturated. And so this can be anywhere from like 1 to 300 milligrams per milliliter. And what is the best concentration to actually use? So in the absolute absence of any other information, go ahead and try to use 10 mg per mil. And I'm going to tell you that you're never in the absence of other information, because you should always do a pre-crystallization trial to identify what the ideal concentration to use is. Okay, so we are not going to really cover a huge amount of information about membrane protein crystallization. It's a somewhat special topic, also very important, and we could run a whole workshop on that. But I do want to just cover a little bit. First of all, that membrane proteins do crystallize. They sometimes crystallize differently. Membrane proteins are typically solubilized with detergents, and one of the key things to keep in mind is that those detergents that are used for purification are often not the same detergents that you're going to need to use for crystallization. When we're thinking about membrane proteins, there's a couple of different kind of common ways that we can pursue crystallization. The first thing is, of course, you start with a purified sample. Just like in any other type of soluble protein, we really need highly pure sample to be able to move forward with crystallization. We can use techniques that are very similar to the soluble type experiments where we add detergents to that membrane protein, and those crystallization screening experiments are going to be very similar to everything else we've talked about. Similarly, we can actually use bicels, so different types of detergent mixtures, detergents and lipids to generate a bicel. Those will also typically proceed using very similar techniques. And then finally, there's a technique called lipidic cubic phase, or LCP. And in LCP, we actually have some additional tools. It's a way that works extremely well for membrane proteins to generate crystals. Okay, so finally, some of the things you want to avoid in crystallization trials. Often, you really want to avoid freeze-thaw cycles of your sample. If you know that your sample is very robust and is perfectly fine undergoing a number of different freeze-thaw cycles, then that's probably fine. But if you're not certain, it's best to try to minimize the number of times that you freeze your sample. Often lyophilization can actually impact the long-term crystallizability of your sample. Okay, finally, let's talk a little bit about some of the things that we recommend to often try to avoid. So first of all, freeze-thaw cycles. So if your sample is perfectly fine and robust when undergoing a variety of freeze-thaw cycles, this is not something you necessarily need to think about. But if you haven't thought about it, then you want to try to avoid too many freeze-thaw cycles. Lyophilizing the sample can have an impact on crystallization propensity. Certainly combining different preps and purifications. So I hope if there's one thing you take out of this portion of the workshop, it's that pure sample is absolutely essential. And in reality, when we take different samples that are from different preps and we combine them, we are making a less homogeneous sample. And then finally, ammonium sulfate precipitation as a purification step. Okay, we've spent quite a bit of time talking about all of the biochemical considerations that you need to be aware of. And in part, that's because this is probably the major thing that kind of sits in the background of whether you're going to be successful in your crystallization trials or not. Okay, so different methods. There are a number of different methods. This phase diagram shows some of them, and we're going to talk about just a couple. The first is microbatch. And so microbatch you can do in two different ways. Microbatch under oil, in which case you add typically a 1-to-1 ratio of your biomolecule and your cocktail, and you put that into a well underneath oil, and then you wait for crystallization to occur. And then batch, in this case, doesn't have oil. You do, again, a 1-to-1 ratio, typically in some type of an Eppendorf tube of your biomolecule and your cocktail. And you wait for crystals to form. Okay. There's a variety of different ways that we can do this in different plates and different setups. The one you're going to hear about quite a bit is our 1536 microassay plate, which we use in the high throughput crystallization center. These are different ways we can set up using both manual and robotic setups. Obviously, the 1536 plate we have to do with robotics. Another very common technique that we use a lot is vapor diffusion. In vapor diffusion, essentially what's happening is we set up a drop mix of your biomolecule and your cocktail. There's either a sitting drop or a hanging drop, different kind of plate platforms. And your cocktail reservoir is going to be at the bottom. And then at the top, either in this sitting drop, is a mixture of your biomolecule and your cocktail. And in the hanging drop, it's a mixture that's hanging upside down in a sealed plate. And in both of these cases, we have a concentration gradient between the reservoir solution and the mixture. And that's going to actually cause essentially concentration in your drop, which will help induce crystallization. And so again, what we're doing is trying to kind of move through that biomolecule-rich saturated phase, hit the nucleation phase, and then generate crystals. I will say that there are some samples that actually do better in hanging drop, in part because you have a surface tension of the drop, and some that do better in sitting drop. So just different parameters to try. Again, there are a lot of different ways we can do this. There's different manual kind of things that we can set up, different manual plates that we can set up, as well as different plates that we can set up for these experiments using robotics. Okay, physical considerations. I'm running out of time, so I'm going to kind of hurry through some of this stuff. Some of the physical considerations we are thinking about are temperature, time, humidity, vibration, gravity. There's a lot of work that's been done really exploring the different physical considerations that help induce crystallization. I will say that some of the common temperatures for crystallization trials are 20 degrees C, 25 degrees C, and 4 degrees C. In the crystallization center, we actually use 23, 14, and 4. There have been some studies actually really looking at what is the impact of temperature kind of on kind of the different types of phase diagram types of setup. So this is actually from a study looking at generating crystals for both crystallography for X-ray diffraction and for neutron diffraction. Okay, this is a really nice example of how the incubation temperature actually can impact the morphology of your crystals. And so what you see here is a set of different crystals that are changing as the temperature is in fact changing. So we have either a needle polymorph or this bipyramidal polymorph, and you can actually see the temperature differential making a difference in exactly how those crystals are growing. This is a parameter you want to be thinking about when you're doing crystallization trials. Okay, time. Crystallization can absolutely take time. This is an example of a sample that we've run in the crystallization center, where you can see that crystals don't actually even start to appear until about week 4. We start seeing a little bit of kind of proto-crystallization,
probably more precipitation and aggregation, but the crystals themselves don't appear until week 4. And so you want to give yourself adequate time for the crystals to actually appear. Again, I'm running out of time, so we will be talking more about crystallization cocktails as we're moving through the day, but I do want to highlight that there are a huge number of different common precipitants that are used in macromolecular crystallization. We use a lot of those in our 1536 screen. We have also done some mining of the PDB to actually look at what different successful crystallization cocktails are available. And so if you're interested in actually looking at kind of the different types of crystallization cocktails that are available in the PDB, we do have this available on Zenodo. And I'm going to just highlight that it's a critically important thing for researchers to include their crystallization conditions in the metadata when you deposit your PDB structures. The reason for that is because if you want to do AI or machine learning or anything like that, you need to have the data. And so here's an example from this project we did where we scraped the PDB and actually pulled details about the types of crystallization components that were in this particular cocktail. But there are a huge number of examples where there is in fact no information included, or the information is so verbose and uninformative that it's like there's no information. So please keep in mind that when you're doing your PDB deposits, you should also make sure to include your crystallization cocktails, as well as all of the other details like temperature, type of experiment, and so on and so forth. Okay, so with that, I'm going to wrap this section up and remind you that our next section is actually on using the National Crystallization Center. Thank you very much. Okay, so for this session of the workshop, recording this, please hold on to any questions.
Sorry, everyone. Sorry, everyone. I had to get that to stop.
All right, Sarah, we've got some questions for you.
Great.
Are you all ready?
I am ready now.
Okay, if any of you want to ask your questions in person, just raise your hand and I will let you, but I will just start with some of the questions that we have in the chat. We have a question from Dana. They're asking, if you don't have access to DLS, is there something else you could use to determine solubility?
That is a great question. And yes, there are a variety of different things that you can do. We're going to talk a little bit about the pre-crystallization trial, and that's one way to kind of really assess the concentration and of your sample and whether it's an appropriate kind of concentration and solubility. You can look at things like your UV vis spectrum, where you're looking at the 280 band, and you can do various things to your protein, try it in different buffers, let it sit on the counter overnight, try to do different freeze thaws of the same sample and see if that 280 band actually decreases, because that would be an indication that that protein is probably coming out of solution. And so there's definitely other ways that you can use to take to kind of get a bead on kind of how soluble your sample is. There are other techniques as well, which, yeah, it would be great to have a whole workshop on that as well. So, okay.
All right. Just something that I wanted to add to that as well. In the past, when I've been working with proteins trying to determine a good solubility buffer, one of the sort of easiest things to do is just centrifuge your sample, take a whole pellet soup for your sample in different sample buffers, and that'll give you a sense of how soluble your protein is in a different set of screens. And so that's something that I think most labs would have access to.
Yeah, that's a great idea, Gabby. So,
all right, a question from Bavita. They're asking, what are the parameters to be considered for co-crystallization?
That is another great question. So co-crystallization is something you can think about, either two proteins that make a complex that we're trying to co-crystallize together, or we've got a protein plus something that binds that we're trying to co-crystallize those two things together. Honestly, some of the things that you need to think about are all of the same things that you are already thinking about for crystallizing your sample. But when you're co-crystallizing, you are adding additional complications, because if you have, say, a two-component mixture, you could have one component crystallize and the other not, or both of them crystallize. And in some cases, you don't necessarily know until you are actually collecting the data whether the thing you have crystallized is your complex or just one component of the complex. There are some different tools that we can use to test those things out. They're a little bit more advanced, but the things that you want to think about in terms of co-crystallization is what are the different concentrations and kind of ratios between part A and part B of your complex. And so those are things that you are typically going to test and look at kind of ahead of time in solution before you're trying to crystallize. And so that's just a brief overview of that. And Gabby, do you have any suggestions on that as well?
I would just mention that I really favor microbatch under oil technique for co-crystallization. I think it works really well. And then, of course, the other consideration, like you mentioned, just being careful with the interpretation of your crystallization results, because it's very easy to crystallize just your ligand or just your protein.
Great.
All right, another question from Bavita. They ask how to optimize crystallization conditions when we see crystal clusters.
So we are going to talk about optimization later in the day. That is not a topic that I talked about in that section. So I'll just answer it really quickly here. I honestly think if your crystals are growing in clusters, that one of the key things that you can try to do is to seed. You can try to break those clusters up and then try to actually force the sample into a different morphology using seeding.
Agreed. All right, Rakesh asks, what is the general method of harvesting a crystal in alcoholic conditions, as they have chances to crack or they sway around?
We are also going to have a session on harvesting. And in terms of harvesting crystals that are in different kind of conditions that are volatile in any way, yes, it's challenging. And one of the things that we really like to do is use the mitogen watershed for harvesting pretty much all of our crystals, because it gives us a little bit of extra time. Again, we're going to talk about some of those things. Gabby, I'm going to let you weigh in if you'd like.
Yeah, I would definitely say hydration is key for especially like volatile conditions like alcohols. So if you can keep the drop as hydrated as possible, that's going to help quite a bit. All right, next question from Oscar. When I try to crystallize my protein, a very rapid precipitation is generated within about 5 minutes, which has a white color. That could be because I have too much concentration.
I'm gonna say very possibly. You may be essentially pushing things kind of too far in terms of your protein concentration, and so if you tamp that back a little bit, I mean, like we've talked about already, when we're trying to crystallize, we're really at this borderline of a phase diagram and kind of solubility. We want to push things into a crystalline form without precipitating it. And so if you're immediately precipitating it, you probably want to lower the concentration. We expect some precipitation to occur, but I would suggest you lower the concentration of that sample. So.
All right, and then our last question here from Ethan. Can you expand on the difference between sitting versus hanging drop? Are there pros and cons to each, or are they different ways of doing the same thing?
They are different ways of doing different things, or similar things. They're both vapor diffusion, and so let's see. Let me go and try to find that slide again, and share it really quickly. Okay, so when we're looking at both sitting drop and hanging drop, we have a cocktail reservoir. And so that cocktail reservoir has the same components as what is in the cocktail drop up above. And in the sitting drop, essentially your sample is sitting inside of a well. And so exchanging with the cocktail reservoir within the sitting kind of space. The hanging drop actually hangs upside down, so you actually have of course, gravity works, so that's great, and that means you have tension here. And so some of the things that we'll sometimes see is that things will crystallize in a hanging drop that won't crystallize in a sitting drop. So the sitting drop, what you're not seeing here is this is supported by plastic, whereas this is actually just hanging kind of over the well. And so that can actually have an impact in whether the sample will actually crystallize or not. So sometimes, like I said, sometimes things will work with sitting drop, but not with hanging drop. And sometimes things will work with hanging drop and not with sitting drop. And we are a big fan in the Crystallization Center as well of using microbatch under oil. And so that's a completely different thing, completely under oil. So, right, I'm going to stop sharing.
And I'll just add on that too. If you have crystals in a sitting drop format that really like to stick to the bottom of your plate, you can overcome that if you can reproduce them in a hanging drop. And then we do have another one real quick from Felicity. How do you image hanging drops, or can it not be done with high throughput?
So we image, so in the Crystallization Center, we do all of our imaging in our Formulatrix rock imagers. And we are able to image sitting drop, hanging drop, LCP, as well as our 1536 platform, which our 1536 platform is pretty unique. I think we're the only place in the world that actually uses this type of platform. And so we definitely have specifics that we have to make use of. So yeah, the hanging drop is not difficult for us to image, and I'm happy to kind of talk with you about how we do that if you're interested, Felicity. So, all right. Well, thank you, everybody. I'm going to turn this over now to Gabby, who is going to talk to us about the High Throughput Crystallization Center.
Great. Thank you, Sarah. Let me see if I can get my technology to work. Okay. Are you seeing the right thing?
Yes.
Good. Thank you. Okay, I'm going to get a little pointer here. So for those of you who don't know me, my name is Gabby Bujaszewski, and I'm the operations manager at the High Throughput Crystallization Center. So it's fairly likely that if you've submitted samples to us before, you've either spoken with me or Tiffany while we're trying to set up your experiment and interpret your results. So I'm just going to talk really quickly today about things that you may already know about the Crystallization Center and maybe some things you don't. So what exactly do we do? So Daria kind of walked us through this macromolecular crystal structure determination pipeline, right? Where we go from our pure protein sample to a model of our 3D coordinates that we can use to make hypotheses about biochemistry. and extrapolate different hypotheses about disease states and how proteins in our body and different biomolecules in our body operate. But one of the major bottlenecks of this protocol is actually crystallizing proteins, right? Because Daria told us it's very challenging sometimes to determine what appropriate crystallization conditions are. Sarah spoke about this in depth as well. And so our role at the National Crystallization Center is to help our users determine an appropriate crystallization hit for their sample. So we do have a high throughput pipeline to determine crystallization conditions. We actually have 1536 conditions in one single microassay plate. So here is a penny for scale. You can see that this is about the same footprint as a 96 well standard tray that you would have in your lab, but this plate actually has 1536 wells. And so what we do with this is we set up your sample in this plate, and then we provide the robotics, the imaging instruments, and the expertise to help you interpret your crystallization results. So this is what our pipeline looks a little bit like. And so there's some things going on behind the scenes. First of all, we start off with our 1536 plates that we coat with 5 microliters of paraffin oil. And we add, previous to setting up every individual experiment, we add 200 nanoliters of our cocktail conditions into these plates. We always take a day 0 image so you can see what the cocktail looks like before we add any of your sample. And this is important because sometimes these crystallization cocktails contain high concentrations of salts and other things that like to crystallize on their own. Sometimes errant sweater fibers end up in these plates. And so you just want to have a good understanding of what the wells look like before you add any of your sample. The next step is where our users actually come in. You ship your purified protein or biomolecular sample to HWI for your setup. And then we will take our SPT LabTech Mosquito and add your sample, so 200 nanoliters of your sample, into these pre-prepared cocktail plates. This takes us about 4 minutes to set up each individual sample. At this point, we then monitor your 1536 wells over time to identify your crystallization conditions. And so you get state-of-the-art imaging. So not only bright field imaging of your wells, but also these 2 techniques I'll talk about a little bit more in depth that can give you a little bit more information about what exactly is happening in these wells, UV-TPEF and SHG imaging. And finally, when you have all of your imaging data, we have Marco Polo, which we will discuss a little bit further in depth. But this is a GUI that's empowered with an image recognition algorithm that you can use to identify your crystal hits without manually going through all of your imaging data. So I want to touch first on this multimodal imaging that I discussed earlier. So in the process of your experiment, you're going to receive 7 sets of visible images. So this is just standard bright field imaging, but you'll also receive UV-TPEF images and SHG images. And together, these techniques are called sonic. And what are the advantages of these 2 techniques? First of all, they can help you verify that what you're seeing in your plate are actually protein crystals, and also help detect submicron crystals that might be invisible to you in the bright field images. So I like to just show a couple sample images to start off with to say, based on just a bright field image, do any of these wells contain protein crystals? So if you look at this first image, it's heavily precipitated, so it's really unclear whether we see any protein crystals at all. The second image clearly has objects in there that are crystalline, as well as this third object. So what can we actually do if the bright field images are ambiguous, like this first image that we see here? So I mentioned this is called sonic imaging, and it stands for second order nonlinear imaging of chiral crystals, which is a bit of a mouthful. So we just go with sonic. And these techniques are UV-TPEF, which is 2 photon excited fluorescence, and second harmonic generation, or SHG. So first of all, positive UV TPEF looks a little bit like this. It has a little bit more of a diffuse signal than what we are going to see with our SHG, but it is being excited in the focal plane as opposed to a UV fluorescence sort of imaging. And so you will sort of see the signal concentrated where you see your protein crystals. The way this is operating is that aromatic residues will fluoresce when exposed to frequency doubled photons. And really, you're getting the most signal in this technique from your tryptophans. And so if your protein lacks any tryptophan residues, your signal might come a little bit from the other aromatics, but really the primary contributor here is going to be your tryptophans. And the amount of signal that you get is correlated with the amount of tryptophan residues you have in your protein. So if you have 1 tryptophan versus 14, there is going to be sort of a difference in the amount of UV TPEF signal that you do have. And so what's actually happening here is that we are exciting with 532 nanometer light. 560 nanometers is actually the tryptophan excitation maximum for 2 photon. So if you have this, so 560 divided by 2, that's actually just your UV 280 that you would expect. And so you're exciting your aromatic residues, and then you're observing fluorescence from these residues in the range of 325 to 400 nanometers, and that is what generates your signal. Related technique is second harmonic generation, and your signal is going to look something like this. This is a very high contrast technique. And so you'll see this very black background, and then your signal is very obvious here with the white crystalline regions. So chiral crystals are going to appear white, and unfortunately, centrosymmetric space groups are invisible, which means that some protein crystals will be SHG disallowed, but the vast majority, about 80% of protein crystal space groups are actually going to be visible with SHG. And so what's physically happening here is that we're exciting with this 1064 nanometer light. And when it encounters a chiral crystal, what's going to happen is that you actually are going to observe your SHG signal at 532 nanometers. And the reason for this is based on the relationship between frequency and wavelength that we kind of know here. So when you're doubling the frequency, you're actually halving the wavelength. And so that's from your two incident photons, and then you observe the halving of the frequency of the light that you're actually shining on your chiral crystals. So this gives us a little bit more information about what exactly is in these wells that I showed you earlier. And so if you look at this first well that looks heavily precipitated here, what you can see is that there's actually signal here for some obscured protein crystals that are positive in both the UV TPEF as well as the SHG signal here. So this is really powerful because just by eye, you would not be able to tell that this well necessarily had any protein crystals to speak of. But actually, this is a significant hit and something that you could take into your own lab and optimize for crystallographic data collection. The second well, which we suspected are protein crystals, indeed, you can see this beautiful UV signal, as well as an SHG signal here as well. And this last one that we saw in the bright field, it has no UV TPEF signal, but it does have a very significant SHG signal. So what we're actually observing here is a salt crystal. And this is something that's relatively common when you're looking at crystallization screens, just because you need such high concentrations of salt in order to achieve the phase change of crystallization. That this is just going to be an artifact of some of these crystallization screens that you need to keep an eye out for. So what does our instrumentation actually look like? We have a suite of robotic liquid handlers and drop setters. And so this is our sort of past generation of instruments, but they're still sort of up and running. And our workhorse instruments for actually setting up our 1536 plate is the SPT LabTech Mosquito. We also have optimization instruments like the Formulator by Formulatrix. We have an NT drop setter. We use our Mosquito drop setter for optimization as well. And then we have an Art Robbins Griffin, and then an Integra for transferring crystallization conditions and plating certain things. So we really do have a suite of instruments here at the HCX Center to help you achieve your crystallization goals. And so those are our drop setters. Oh, here we go. This is actually a video. But we also have a suite of imaging equipment provided by Formulatrix. And so we have 2 of these Rock Imager 1000s, and these imagers are equipped with the sonic functionality. We also have, Sarah mentioned this mitogen watershed. So this is really good for humidity controlled harvesting. It gives you a little bit of extra time in the drop when you're trying to loop things, or with these sort of like volatile alcohols and things like that. It'll just give you a little bit more lead time before they evaporate. And of course, we have all the other standard crystallization equipment like pucks and shipping doers here as well. So how do we deal with so many images? If you've ever done an experiment with us, you're constantly receiving emails saying that your imaging is ready. Here's how you access it. And so ultimately, we end up delivering you about 14,000 images per experiment. And so this is quite a significant amount of data. And so we had asked, is there a way to apply machine learning or artificial intelligence to help score images? And this is something that not only us, but also different pharmaceutical companies were sort of really interested in figuring out if it were possible to do. So many of you have probably seen this meme showing how similar chihuahuas and blueberry muffins look at just a cursory glance. And of course, we can take the time to go individually and say, okay, muffin, dog, dog. But this is kind of time consuming, and our task would be a lot easier if we could train an algorithm to identify the differences between puppies and muffins by training an algorithm on labeled images of either puppies or muffins. So our problem here at the Crystallization Center is similar. How can we apply machine learning algorithms to help us identify what are protein crystals and what are not, without necessarily having human intervention every single time? So the machine learning tool that we've built to sort of address this is reliant on what's called the Marco algorithm. So this stands for Machine Recognition of Crystallization Outcomes. And so this algorithm was initially trained on about 500,000 labeled images. And so here's the training set size, and there was a small 50,000 image test set. And it was trained on different crystallization methods, so batch and vapor diffusion, as well as images from many different sources to try and overcome different artifacts that the algorithm would sort of obtain by being trained on only images from one specific source. So the scoring algorithm helps to sort through images really rapidly to take the time off of the experimenter. And this was a collaboration with Google Brain and other crystallization centers and pharma companies as well. And so what's going to happen is that the algorithm will analyze your image, and specifically for this image, Marco determined that there's a 0.93 probability that there's a crystal here. So if we zoom in here, we can see actually there are 4 crystals sort of clustered around here. That would be sort of difficult to identify if you were trying to do this manually and sorting through 14,000 images. So how does this exactly work? Marco is a computational neural network. So it's a stack of nonlinear filters. There are 3 filters here. And then at the top of these filters is the classifier model that's operating on this feature map that's sort of generated by the filters. It's using the Inception v3 image recognition architecture. And it identifies whether your image is crystal, precipitate, clear, or there's an other category as well. So what does the performance of Marco actually look like? About 94% of the test images from that test set that we discussed can be correctly labeled, which is a really high success rate. The limitations, however, it only uses bright field images. So this is not current with Sonic, so UV and SHG that I discussed earlier. And it's sort of difficult to update the trained model with additional data. So Ethan Holloman, who was a student here for a little bit, actually incorporated Marco into a GUI that we actually use for analyzing our crystallization data. And so the GUI is something that Elizabeth is going to talk about right after this. But it's well set up to show you your bright field and sonic images. You can sort of scroll through them. You can classify them with the Marco algorithm and only look through the hits. But you can also directly correlate this to your crystallization conditions so that you can sort of rapidly get ready to optimize the hits that you see in your crystallization tray from us. So finally, I just want to end with sort of logistics about who can use the center and how do you use the center. So who are our users? Honestly, anyone and everyone. But you would be an excellent candidate to use our services if you've already tried all the screens in your lab and sort of had mixed success or less success than you were hoping for. If you manually set up all your trays, like I said, we can do this in 4 minutes. You manually inspect all of your wells for crystal growth. If you do this, I know I've done it in my past. It's very time consuming. It takes, depending on how many trays you have, it can take half a day. If you don't have advanced robotics or imaging, if you need fast, easy access to screening a wide range of parameter space, and like I said, anyone can use our services. And then in the past year alone, we've worked with over 200 samples from 125 distinct users and 69 different research groups. And so we have a lot of experience working with investigators from almost any situation that you can imagine. So please do reach out if you have any questions about that. So how do you actually access the High Throughput Crystallization Center? Or the best way to figure that out is to go to our website, getacrystal.org. Every month we post the upcoming dates. And so we need to sort of not have our pipeline operating continuously because we do make our plates fresh every month. And the reason for that is that if we make them too far in advance, the drops do dehydrate. So we make them fresh for you every month. And this is based on a reservation that you make with us. So if you email htslab@hwi.buffalo.edu and let us know that you would like a plate for the upcoming run and just let us know when you would like to send your sample, that's the best way to do it. Some frequently asked questions that we have here, sample requirements. We're requiring 400 microliters of pure concentrated sample right now. This gives you a little bit of information about how pure your sample should be. So these are things that Sarah discussed. Sorry, this number is old. We only need 400 microliters of your concentrated sample. The suggested concentration is 10 mg per mil, but if you have a better sense of how your biomolecule is operating, you can send it to us at a different concentration as well. So shipping and sample setup, we usually ask that you ship Monday through Wednesday, and this is just so that we receive your sample during the week and it's not sitting here over the weekend, sort of thawing in our loading dock. Priority overnight is the best way to ship it. There is a sample submission form and includes some identifying information. We really appreciate that, so we don't have to try and hunt down who sent us samples. And then emailing us is always your best bet. Send us tracking info. This is usually what our tables look like in the crystallization center on the first day of the run. So you can see it can get kind of overwhelming. And so if you can help us in any way, we certainly appreciate that. The last thing that I wanted to mention is something that we've recently, within the last year, sort of launched, which is our Meritorious Users Program. And so this is specifically for investigators at PUIs, MSIs, or under-resourced labs. Essentially what you get is this entire screening setup that I have just discussed, but we subsidize it for these specific users. And so if you fill out an application, we are going to be doing or awarding these about twice a year. So if you apply, we will look through your application and we will award up to 3 plates, 3 discounted plates per lab. So I just wanted to thank you guys for listening. Our team is listed here, and I'd be happy to take any questions.
All right, so Gabby, we've got a couple of questions for you.
Yes.
Um,
Okay.
First off, Dana, do you want to unmute and ask your question? Okay, I'll read it. Is Marco able to be used with all imagers or only certain file types from specific imagers?
Okay, so it is translatable. Sarah, you actually probably have more information about this than I do.
I'm
But
happy to answer
yes.
if you want.
Yeah, you can give a little bit more specifics here.
Okay, so yes, Marco is actually was trained on a large number of different types of images, and so it can be used with all different types of imagers and different file types. The algorithm itself is open source, and the data are available. Part of the reason that we implemented the Marco Polo GUI was because the algorithm was not necessarily easily implementable. And so it's a very great tool, and it does a really good job, but we wanted to be able to easily implement it for our 1536 plates. So our Marco Polo software GUI right now is really designed for the 1536 plates. We are in the process of trying to expand the GUI so that actually you could use virtually any type of plate as long as you have a plate definition file. But that's work in progress right now. So I hope I answered your question, and no problem for not answering, not reading your question out loud. Let's go to the next question here. Will protein precipitation have UV-TPF signals? If yes, how can they be distinguished from UV-TPF signals from crystals?
That's a great question. So yes, precipitation will have UV-TPEF signals, and it can be sort of a complicating factor. I would say the best way to sort of interpret this specific situation is to see if your crystals also have an SHG signal. So interpreting both the UV and the SHG together is sort of the best way to figure out, okay, with the UV, is it protein, right? And then with the SHG, is it crystalline?
And I will also say that if you just have UV, any type of UV, you will similarly see essentially any type of aggregated sample. And so that's crystal, that's aggregation, that's phase separation. You will often see signals from that. There are various dyes that you can use to like add to your crystal sample. This is something you could do in your home lab to assess, is this actually crystalline or is this precipitate? So, okay, Francisco, I'm going to read your question. I'm not sure if I follow it. So, so perhaps you might have to unmute to clarify. I have used Marco to visualize the samples evaluated in the National Crystallization Center, but we can see them in the other wavelengths. Are they available in some way? So
I'm
you
wondering
can't,
if
oh, you can't see them.
can't see. So I think they might be saying they can't see the sonic images.
So you should be able to see the sonic and the UV-TPF images in Marco Polo. And if you're having trouble with that, I'm going to encourage you to stay into the next section where we're going to actually be doing a little bit of a tutorial on Marco Polo. I will say that Marco itself was not trained on SHG or UVTPF. And so if you run the Marco algorithm on those types of imaging modalities, it will fail catastrophically. So just don't do it. And again, that's another thing that we are working on kind of in a long-term way to try to update and be able to make use of things. So, okay, Gabby, a few more questions and then we'll turn it over to Elizabeth. Okay, Felicity, what is the smallest crystal that the sonic technique can see? E.g., would it do microcrystal slurries or other very small crystals among precipitate?
So this is a question that we're very interested in here at the National Crystallization Center because we have a strong interest in microcrystal techniques. So I think the official sort of party line on this is that we can see down to a micron. I think it's probably capable of seeing things in the range of like 700 to 900 nanometers. Below that, you will probably need some sort of adjuvant, like a some sort of dye or something to see things that are smaller than that. But certainly you'll see a microcrystal slurry and other small microcrystals.
Yes, and Felicity, this is something that we're actively working on in kind of our when we put our research hat on, and definitely have are really working on pushing ways to actually visualize the super small crystals for, micro ED, for X-FELs, and for other things. And then the final question, also from Felicity, how many repetitions of each condition do you do in your 1536 plate?
So there really is not a lot of replication of anything in the plate. I would say there are conditions that are quite similar to one another, but all the conditions are different enough from one another to be considered individual conditions.
All right. Well, let's thank Gabby for a very fantastic presentation. Thank you. And next up, I realized that I didn't actually introduce myself, and I really apologize. Anyway, I'm the director of the National Crystallization Center, and I work here at HWI. Okay, next up is going to be Elizabeth. So Elizabeth Snell is a research technician in the Crystallization Center. She does a lot of work both in the Crystallization Center and also on our microcrystal projects. And she's going to start us off with doing a little tutorial on Marco Polo. And then for anybody who is interested, we're going to do some breakout rooms. I know we're running a little bit behind schedule, but this is running right up against lunch. And so our lunch will then go until 1:30 Eastern. And so if you are not interested in Marco Polo, you can go have lunch. So, okay, take it away, Elizabeth.
Hi, I'm going to give a brief tutorial about Marco Polo. And what I'm going to do is just to walk you through using a data set that we have uploaded to our FTP site, and you should have gotten an email about that yesterday. So here we go. Let's see. Is that up? Can you see that okay?
Yep, that looks good.
Okay. So I have already loaded into Polo the most of the images from the data set that we shared, but I will go over how to import these images into Polo briefly. We do recommend for the data sets that you use an application called FileZilla. It is usually much more successful than the Marco Polo app itself, but I will show you briefly how you can import these things from the FTP site. Just so you go to import and images and from FTP. I'm not actually going to do this, but I will show you how you would do it. You would input your username and your password and connect, and then all of the files that you have with the National Crystallization Center will show up, and you can select which ones you want to download that way. But again, we do recommend that you use FileZilla. Once you have those images locally, you can import them in two ways. You can actually drag and drop the files into the sample section over here, or you can go to import images, and what you get when you use FileZilla is the RAR archives. So we'll do that. And so you open this importer and you're going to browse for the RAR, and you want to look for the folder where you have downloaded the images from the FTP site. And once you have done that, you select the one that you want to import, and you select it and then click on open, and it will bring it into the Polo app, hopefully. I don't know why it's not working. Well, I'll tell you what would normally happen is the file would show up here, and you would select it and then click on import run, and that will pull it into the app. The other way of doing it, like I said, is just to drag and drop the file into the sample window. I'm just going to close this and hopefully continue working.
So I'll take this moment, Elizabeth, to just say
Okay,
that
please do.
that Marco Polo, our graphical user interface, is built by a really wonderful post-baccalaureate student, but it's software that we've built in-house, and so there's definitely a little bugginess sometimes. So if you encounter bugs, please email us, because we will definitely like to hear about that, and it'll help us troubleshoot, especially as different operating systems get updated. It helps us to know what's going wrong so that we can try to fix it.
Okay.
Are you paused now? Okay.
I don't know what it is doing. I think, oh, it's still working. I just don't really have a cursor, but we'll just work with what we have. So once everything has been loaded in, you'll see all of your choices for what you want to look at in the sample window. And what we're going to look at, I've already loaded it in, and it is this, it shows you the date and the type of images that you're looking at. In this case, we're looking at images from the 4th of April and the visible images. And the first thing we're going to do, because it does take a few minutes, is to classify the run using the Marco algorithm. And what you would do is you would select which data set, which image set you want to classify and select it, and then just click on classify selected run. And that will run in the background, and you can look at your images while that is running. Because it does take a little while, I've already done that. And you can see when you look over on the right-hand side, if you look in the image details, it shows what Marco has classified the image as. And this is an example. Occasionally Marco does get confused as to what it's looking at. You can see clearly that this is a bunch of crystals here, but there are so many of them that it looks like precipitate. So it's been classified as precipitate, but the confidence is fairly low. So this just shows that, you know, sometimes it's a good idea to look through the images yourself, just to make sure that Marco has classified things the same way that you would. So to move through the images, you can use the arrow keys on your computer, and it will take you sequentially by well number through the images. And you can see we have a lot of crystals and then some precipitate and so on and so forth. You can also navigate by clicking on the previous and the next buttons. And there's another good way of looking at your images. You can look at all of the dates, and that will show all of the dates from all of these files. So if I click on that, so it shows you the progression of crystallization in this well, for example. You can also click on show all spectra, which will show you this is the SHG over here. This is lysozyme, and lysozyme unfortunately doesn't have SHG signal in most of the crystal forms. So this is the SHG. It's clearly crystalline, and you can see that in the UV. And if you want to go through and score the images yourself, there are a couple ways you can do this. You can click on these classification buttons here. You can have crystals classified as crystals, and it will automatically move on to the next well. If you go back to the previous one, you can see that it has now updated under the image details that you have scored this particular image as crystals. And you can also use number keys. Number 1 corresponds to crystals. Number 2 is for a clear well. Number 3 is for precipitate, and number 4 is for others, such as phase separation. There is a useful way of looking at these images. You can filter and only look at, for example, the crystalline, the wells that contain crystals. As I haven't gone through and scored this entire data set, so for the classifier, we would select Marco. So if we want to look at just crystals from Marco, you click those 2 things and submit filters, and it will pull up all of the wells that contain crystals that have been scored, wells that have been scored as crystals by the Marco algorithm. And then you can just go through those the same way you would go through all of the wells, using either the arrow keys or the previous or next buttons. You can also sort your images. You can sort them by one of 3 things. You can sort them by Marco confidence level. You can sort by well number, which is the default, or you can sort by cocktail number. By cocktail number. In addition to being able to view these things here, you could also use the export function, which is located up here, and there are 4 options for exporting your data. You can export it as a comma separated value file, which is compatible with things like Excel and Google Sheets. The JSON, which is JavaScript object notation, which is you can view that with text editors. There's also an MSO, which is actually just basically a legacy file type that we used in the Crystallization Center, or you can export them as a PowerPoint file. And PowerPoint can be really useful because it can save all of your images. It can save your SHG and UV images for a particular, and the bright field for a particular well. You can also save the run once you've categorized it by Marco by going to File and Save Run, and you can either just save it with the default name or you can save run as. And this will save it as a .xtl file, XTAL file. And if you come back to it later and you want to open it, you can import images from a saved run, and just from wherever you had saved it, you can select that. The last thing I wanted to talk about is a quick way of looking through your images. And I'm just going to go back and deselect these things. And if you go on Plate Viewer, this will, in order to view the first one, you have to go to the next image, either by using the arrow key or the next button here, and then you can go back to previous. And what that does is it shows you 16 wells at a time, and it is showing you 16 wells based on the way that they are set up in the plate. So if you can, I don't know if you can see this, but it gives you information such as here it's giving you the well number and the human classification. You can switch that and make it Marco classification. The color also tells you what it was classified as, and you can see that under image coloring. So anything that was classified as crystals is red, clear is blue, precipitate is green, and other is orange. This is showing you wells 1, 2, 3, and 4, and then 49, 50, 51, and 52, because it corresponds to the physical location in the plate. But you can go through fairly quickly this way if you're just wanting to scan rather than look at each individual well. So that's about it that I had. I don't know if there are any questions. We
Great.
can.
I think that a good job, Elizabeth. Thank you
Thank
so
you.
much. I think there was one question about the basis of the Marco confidence, and Gabby has answered that already. And then there is a question from Francisco about how he's opening his files. And I think that is part of what I think will work really well in breakout rooms. So I am going to attempt to set up two breakout rooms. Does anyone have any general questions about our Marco Polo GUI before we do this? And otherwise, we will see anyone else back for lunch after lunch at 1:30 p.m. for our next set. All right, so we will just kind of do a little bit of basic introduction again here at the second half of the crystallization workshop. And Again, thank you all for coming. I'm going to actually run through our introduction slides again in case there's anybody who has joined us for the second half who wasn't here for the first half. And so let me just minimize here. And so we are recording this workshop, and these slides and videos will be posted, available probably in the next week or two. In the second half of the workshop, we're going to be talking a little bit about optimization and harvesting, and then we're going to have presentations from folks at SSRL, CHESS, and NSLS2. And then we will have time for a final Q&A and wrap up. And of course, if you have questions, you can always send us an email to kind of inquire about how to help with your crystallization needs. Again, we're located in Buffalo, New York. Here is HWI in downtown Buffalo. And the people who have been participating in this workshop, myself, the director of the National Crystallization Center, Gabby Budziszewski, who is the operations manager in the Crystallization Center, Chris Campomizzi, who is a postdoc here at HWI, working with Miranda Lynch and I. Miranda Lynch, who's a collaborator and also works with the Crystallization Center, and Elizabeth Snell and Tiffany Wright, who are research associates in the Crystallization Center. So thank you guys for all of your hard work. Thank you as well, again, to our speakers. We have a team from SSRL. We're going to hear from Sylvia, I believe, this afternoon, Vivian from NSLS2, and Jenny from CHESS. And of course, thank you to our sponsors. We would not be able to do this type of workshop without our sponsors. And so to the NIH for funding our national resource, as well as Hampton Research, Mitogen, Formulatrix, and SPT LabTech. Thank you so much for your support of this workshop. All right, I'm gonna stop sharing that. And this next is also pre-recorded. And so I'm just making sure I have it queued up and ready to go. Okay, so first section, we're going to talk about optimization. Okay, so for the optimization section, I am also pre-recording this. And so we're just going to dive right in. We're going to talk a little bit about some of the different things we think about in terms of optimization. And so you have a crystal hit. This is great. You're very excited. You've seen it. You've imaged it. What next? Okay, so if we think about, again, go back to our original diagram where we are screening different crystallization conditions to generate a crystal. And then our goal is to kind of really move through this whole structure where we collect data, we're able to work with the data and solve our structure. And so in an ideal world, what that would mean is you're going to take that crystal that you found, harvest it, find the best cryoprotectant on the first try without having to try a bunch of other things. Your crystal is going to diffract beautifully, and you're going to get a full data set and solve your structure. And really, you get all the information that you possibly need from that particular single structure. I'm going to say that this is rare because often you're going to have to actually do a bunch of iterative steps. We use the information from that hit that we found to kind of further the experiment. And so we take this and we iterate through the different steps. And so we have a protein crystal now, and then we're going to actually go through and work through to try to kind of really get very good high resolution data. Okay, so why do we optimize? Well, first of all, both crystal size and perfection play a role in getting a data set that leads to a structure. So
Perfection doesn't always relate to physical appearance for crystals. You can have the most beautiful crystal in the world and it can diffract poorly. And similarly, you can have things that really just are not that beautiful and they can diffract beautifully. And so it really, is very dependent on the crystal. So typically a hit from a screen is going to be your starting point. We use hits from the high throughput screen. We'll talk about how we optimize from the high throughput screen shortly. But when you optimize it, essentially means really changing a variety of the different variables that we've already talked about: precipitant, concentration, protein concentration, temperature. And a lot of the variables can actually be very dependent on one another. So you want to keep in mind that our goal is, in most applications, that we're going to have to be looking for a three-dimensional crystal that's got at least some dimensionality in kind of each size, and that you actually need that crystal growth to have occurred consistently and well over all three of those dimensions. Okay, so how do you actually optimize? So the first step is you have to identify your parameters. So you need to know what your protein concentration was, what the cocktail was that induced crystallization, what your initial protein cocktail ratio was, because that will likely change. What the temperature was. Did you have cofactors? Do you want cofactors, ligands, binders? What is the method of crystallization? Do the hits that you've found have anything in common? So are there a particular kind of range of peg sizes or pH or salt? Are there different additives that have been helpful in the crystallization? That can be important. If you always have some type of a magnesium salt, that might be indicative that you actually need magnesium to have a very stable sample. And finally, what is your cryoprotectant? We will talk about cryoprotectant briefly here and then a little bit more in the next section. So one of the goals that we highlight is that when you're optimizing, you want to be diversifying the hits that you choose to follow if you have a lot of different options. Now, if you've only got one crystal hit, you need to follow that crystal hit. But if you've got 20, choose 4 that are different from one another, because that will give you the most possibility of having, for instance, data in different space groups. And all of these data may provide different and relevant information for your biological system. So often we think about the first thing that we do in optimizing is devise a grid optimization. So we will typically take the conditions that have been successful. So say your precipitant and your pH, perhaps PEG, ammonium sulfate, lithium chloride, and we're going to create a grid where we scan across different parameter space. So you can see in this example that we have a variety of pHs on one dimension and a variety of salt concentrations on the second dimension. And we can do a variety of different things with our different grid screens where we're creating an optimization space. So this is an example showing, in this case, NaCl on the top and pH on the side. And you can see that there's a space where the crystals are growing, and that the crystal morphology actually changes as kind of the parameters change. And this is a way you can change multiple parameters in one grid so that you can kind of explore what the best things to do are. In this case, we're looking at varying PEG along one dimension and additive along a second dimension. What you're seeing here is day 0. There are no crystals here. After 8 days, you can see that we do have crystals forming, for instance, in this well, down here. And so again, modifying and changing what's happening is really important because you're not exactly sure where your crystals are going to form. So when you're designing a grid screen, the first thing that we try to do is center your hit condition. And so what does that mean? You try to test the conditions that are kind of above and below where you hit in your initial screen. And you try to include at least one well that exactly matches that initial hit. And so you have an initial hit and you're going to screen around it, both above and below, in different parameter spaces. So PEG, pH, additive. And you want to start off relatively broad, and then in the next step do narrow as you see different crystallization. As you saw in the previous example, there's only a few of those kind of conditions that actually are giving crystals. And so if we need to iterate through and do another step, we may just want to optimize further around those conditions. And so you might choose a broad grid screen and then a more narrow grid screen. So how many conditions should you optimize? So ideally, you will have actually multiple hits to work with as starting conditions. And then the best thing is to identify common and uncommon components. So if you have multiple conditions that contain, again, some type of lithium salt, you probably want to include a few of your choices in optimization as including lithium salts. But if you have a few that do not contain lithium salts, then you might want to also use those. We will typically choose 4 distinct different conditions to optimize in parallel, and we will also vary the protein cocktail ratios in optimization. So what does this look like? So I'm showing an example of a 1536 plate where we've had 8 hits that we've identified that we would like to optimize. So what we would do in this case would be to select 4 different distinct conditions, and we would create 4 different 24 well grid screens in a single 96 well plate, and we would also vary our drop volume ratio. So keep in mind we're going from micro batch under oil here to vapor diffusion. So we can also go directly to micro batch under oil, but we often will actually start by doing vapor diffusion. We could talk about why that is if anyone's interested. But what you can see here is the 4 different colors represent the 4 different grid screens around 4 of these 8 conditions that we've chosen to optimize. Another thing that we do when we're doing optimization is seeding. So seeding is an incredibly wonderful way to essentially start with things that have crystallized, even if they haven't crystallized well enough to diffract or they're not large enough. It essentially starts at the nucleation step instead of at the having to move through that saturated phase step. And there's a variety of different ways we can transfer to a drop in the metastable zone to be able to get macro seeding larger crystals. We can actually disrupt those starter crystals and transfer those into a metastable zone to get better crystals. We can also do streak seeding. So using a cat whisker or different tools, we can actually seed again into the metastable zone to be able to get better crystals. And so seeding is an incredibly important tool for us to make use of when doing optimization. So we then iterate. We typically will have to iterate based on crystal quality, based on diffraction quality, based on how the crystals are behaving when we optimize, when we harvest, when we put them in the beam. And so we take those initial hits and we iterate through these things. What are the different types of diffraction data that can give us information about how our crystals are behaving? So first of all, this is an example of an ideal case where we have well-defined spots, a distribution of both intense and weak spots, and there's a single lattice in this sample. There are a lot of things that can go wrong when you're doing crystallography and taking diffraction data, and these are some of the pathologies that you might see when you're collecting data. So you can have multiple lattices, you can have highly mosaic data. If you have highly mosaic data, this is going to be very difficult to work with. You can have disorder in one direction. So in one direction, it can look beautiful, and in the other direction, it can look actually really very difficult. There's different crystal pathologies that can occur when the crystals are growing, and anything that's going to break your crystallographic symmetry will impact your overall data quality. And if you are impacting your data quality, that has an impact on whether you can actually solve your structure. All of these different things can be caused by crystal growth, cryoprotection, soaking things in, leaving stuff out that you need. And so these are all the different things that we have to think about as we're iterating through our optimization steps. Twinning is a very particular difficulty, and is one that, frankly, when we find twinned crystals, we typically will actually take a step back and try to find a different condition, because it's actually very difficult to work with conditions that are generating twinned crystals to generate non-twinned crystals. And again, sometimes things will look absolutely beautiful, and they're actually twinned, and sometimes they will actually not look beautiful, but they will diffract beautifully. Finally, mosaicity is another thing that we have to think about as we're iterating through our optimization steps. So essentially, all crystals are going to have some mosaicity. It's a measure of the spread of kind of the different orientation of your crystals. And so you can kind of see that in this box here, where if you have some slight distortions, that's going to impact your diffraction data. These things can be impacted by how the crystals are being handled, how the crystals are growing, all of these kind of things. And so mosaicity is another factor that will feed into the iterations of how you're optimizing your crystals and the steps you're taking. So again, what we're going to do is iterate based on crystal quality, diffraction, data. We're going to keep iterating. And I want to really highlight again that multiple crystals may provide different space groups, packing, ligand binding, etc. So if you have multiple different conditions, use multiple conditions and do those in parallel. It will help you in the end. And finally, there are definitely other factors to consider in terms of how we're
optimizing and what our end data are looking like.
Okay. Well, I'm just going to make sure that that's turned off so it doesn't start over again. Does anyone have any questions on kind of our optimization pipelines and protocols? And how we're doing things? Claudio, I see your hand.
Thank you. I'm sorry if I lost the first part of our talk. I was doing German class. I hope I didn't miss anything important. Concerning this talk, we did a grid screen because out of 8 plates, only one well showed some very small crystals. So we did a grid screen optimizing pH, peg nature, and concentration, and we found many crystals. Then we decreased the concentration of protein, hoping to find less bigger crystals. But what we found was even more and even smaller crystals.
Yes.
How?
Well, so if you think about kind of how if you think about the phase diagram, again, your concentration of your protein is going to impact the nucleation that's happening, and that will then impact what the size of your crystal is. It is a lot of trial and error, though. And so are you having the problem where you are able to get crystals, but they are too small or they're too big?
They are too small.
Okay.
And I wouldn't say therefore, but when we measure them, they haven't diffracted well.
Okay. What temperature are you growing your crystals at?
20 degrees.
Because one of the things you might want to do is think about cooling it down a little bit, because that might slow the nucleation and the crystal growth process, and that might help it grow more regularly. If that makes sense.
Yes.
Okay. And the other thing that did you try to diffract a bunch of different conditions? Okay.
Yeah.
Okay. Yeah, because sometimes
We
yeah.
out of different ligands, if even different nature of ligands, so fatty acids and compounds, nothing worked. Yeah.
Sounds like this might be a good candidate for either macro seeding or streak seeding, since you can
Yeah.
already make small crystals.
My advisor suggested me that microseeding with the whisk, and that's something that we have to do next. It's just that we are a little bit in time trouble.
Yes. I will also say that one of the things you can do when you're seeding is actually seed into, it's essentially a random matrix mix seeding. So you can seed into completely different conditions, and that will sometimes essentially find conditions that will be very good for growing crystals, as long as you already have the nucleation points. And so I strongly recommend that as well. So.
All right, so we've got a question from Thatmi. They ask, what could be the possible explanation to have fewer spots in diffraction data?
As in, I think I'm not understanding your question.
Thatmi, if you
le-
could elaborate a little bit, are you talking about few high resolution spots, or are you comparing two data sets and seeing fewer spots in one versus the other? Let us know.
Oh, jeez.
Hello.
I thought me.
Something happened.
Okay, we can hear you.
Yeah. Hello. I hope you can hear me.
Yep.
Yes.
Okay. So I was. We have got the crystal. Then we did some X-ray crystallography, and in the diffraction data, we are getting like few spots of those crystals, and then those crystals were small also. So I was thinking, what could be the optimization that I could get to optimize that work?
Yeah, so if you've got very small crystals and so you have weak diffraction because of that, you might want to try to get larger crystals, or use a microfocus beamline on the smaller crystals. So those are some options. And again, I strongly recommend seeding when you have things that are not working well in terms of the diffraction quality, or trying to find another condition. But to me, the easiest way to find another condition is using seeding if you've already got some type of crystal that has formed. So I definitely strongly recommend that.
Yep. I would also say if there are really very, very few diffraction spots, it could be salt. All right, next question from VJ. What is the suggestion for optimization of a protein with two forms, oxidized and reduced? You'll love this question, Sarah.
Yeah, so we think about this a lot in some of the work we do with metalloproteins in our research lab. So that, and it's a great question because it can be extremely difficult to get if you want a strict oxidized sample and a strict reduced sample. So typically you're looking at things that have some type of metal. And so the thing that I strongly recommend to do if you're, first of all, trying to even determine what oxidation state you're working with is to actually go to SSRL and use Beamline 9.2 at SSRL, because they have UV-Vis microspec. And so if you have a signal that that will give you an ability to determine whether you're working with an oxidized or a reduced sample. You can start to tell what are the different things that are generating and helping your crystals and your sample to be in one or the other of those states. And so you are going to hear a little bit about SSRL in some more specifics in just a couple of minutes. And I will say, feel free to reach out if you'd like to talk more about this. It's, I think, a really big topic. If you're trying to understand oxidized versus reduced, it can be very tricky. So.
All right, Justin is then asking about cryoprotectants. So Justin, I'm actually going to talk about this quite in depth in the next talk. So if you could save your question for the next one, hopefully I'll be able to address it and we can have a chat about it. But then we have a question from Rakesh who asks, sometimes macro seeding produces some small satellite crystals apart from the large crystal. How can we avoid it?
Wow, that's a great question that, you know, I'm not actually sure, because essentially you're pulling your nucleating crystals through through the conditions that you're trying to kind of get new crystals to grow in. And so sometimes you will definitely get larger crystals, but it's a feature that you also get some little guys on the side. And Gabby, have you ever been able to kind of avoid those when you're seeding?
No, not, I mean, this isn't something that I've necessarily encountered. Some thoughts that I have on this are, when you're going to harvest, you could actually try and chip those little crystals off if possible. Sometimes it'll affect the quality of your larger crystal, but sometimes even if you scratch it or crack it a little bit, it'll be okay. The other thing that we've sort of tried for preventing crystals from attaching to the bottom of a well is coating with a substance called fluorinert. And I'm almost wondering if that might help. I have no evidence for this, but I'll type it in the chat. You could try it.
Yeah, it's an idea. All right. Well, if there's no more questions for this session, we're going to turn it directly over to Gabby, Gabby and Tiffany possibly, to talk about harvesting.
All right. Okay. Can you see the right screen? All good? Okay. So I'm just going to continue our conversation about harvesting and talk a little bit more about cryoprotecting and harvesting crystals. So another thing that I wanted to mention, going off of kind of what Sarah had talked about, is you really kind of need to understand the mechanism of action of some of the components of your crystallization cocktails. And this is really going to benefit you when you're thinking about your specific circumstance with your protein. So for example, salts as precipitating agents, they have a linear impact on how well they function as precipitating agents. And so anions typically follow the Hofmeister series for effective precipitation of proteins when this condition is met, when the pH of your crystallization condition is greater than the PI of your protein. And so if we kind of think a little bit about PI and pH, when your pH is greater than your PI, you're sort of on this blue end of the spectrum here, where the negatively charged form of your protein is going to predominate. And so the anions actually have the same charge as your protein in this circumstance. And so they're going to follow this series. And then the reverse is true as well. When the pH of your crystallization condition is less than your PI, so it meets the condition where it is positively charged, the effectiveness is the reversed Hofmeister series. So it starts on this end and goes this way as far as effectiveness of salts as precipitating agents. And the reason for that is because in this case, your anions have the opposite charge as the overall charge of your protein. Non-chaotropic salts, so these are the salts that we use for salt precipitation or salting out. They preferentially bind water and they mask charges between proteins. So what this is kind of going to do is it's going to remove water molecules from being able to be accessed by your proteins and sort of push them together so that they're forced to favor these less favorable intermolecular interactions between proteins versus interacting with solvent. So polymers, these are things like PEGs and glycerols and things like this. They have a logarithmic impact on their effectiveness as precipitating agents. And their mechanism is it kind of depends on the molecular weight of your polymer here. But in general, they trap water through interacting with the polymer and they make it unavailable to the protein. So again, it's inducing this sort of intermolecular interaction between your protein molecules versus interacting with solvent. Low molecular weight PEGs act as good cryoprotectants, and high molecular weight PEGs likely induce molecular crowding, so kind of forcing your protein molecules together. MPD is kind of a midway reagent, so it acts sort of like a PEG, an organic solvent here. It lowers the chemical activity of water, so the ability to sort of hydrogen bond to your protein, and reduces electrostatic screening of the solvent. And so your proteins are more likely to be able to participate in electrostatic interactions with one another. And so you might imagine this is actually particularly effective for nucleic acid crystallization, and nucleic acids interacting with proteins specifically really depends on a lot of electrostatic interactions. So it makes a lot of sense if you think about the system. There's also a good relationship between understanding pH of crystallization and the PI of your protein. So this is a chart showing pH minus PI, so effectively the difference between your pH and PI. So you have positive values and negative values here, and the PI of the proteins. This is from a paper in 2004, and the general rule here is that acidic proteins tend to crystallize a little bit above their PI, whereas basic proteins tend to crystallize anywhere from like 0.5 to 3 pH units below their PI. And so that's reflected here with these boxes on the diagram. And so what you also want to kind of be thinking about is the surface of your protein and what sort of amino acids are available for making these intermolecular contacts. And so some strategies that I've sort of seen to introduce crystallization from one of our users actually resurfaces a lot of the surface amino acids to arginine. And so that's to sort of improve these intermolecular interactions and the crystal lattice. And so all these amino acids, arginine, lysine, histidine, glutamine, and aspartic acid, they are affected by pH. And so their protonation state is dependent on the pH of your cocktail. But importantly, also the amino groups and the carboxyl groups of your peptide chain are also going to be affected by pH. So this is something that you need to be thinking about. Just back to when you're considering what sort of method you should choose for optimizing your hit conditions, vapor diffusion, Sarah mentioned, we usually start with vapor diffusion. And this is kind of purely for the reason that the way that our whole optimization protocol is set up. So for vapor diffusion, you need a significant amount of crystallization cocktail to serve as your reservoir. And so we actually use the reservoir that's plated into these plates then to set up our drops. And so the drop is set up with lower precipitant in the drop than the reservoir. And the reason why is because you're diluting this cocktail in the reservoir by half, potentially with your protein. The mechanism is that water is lost to the reservoir because of this lower precipitant concentration in the drop until equilibrium is actually achieved. And microbatch under oil operates in a little bit different mechanism. So we actually achieve the final concentration of the drop upon mixing, so upon setting up the protocol. And so the reason why this is different is that we need to be starting in the nucleation zone for a batch experiment to be productive. Whereas with vapor diffusion, we often start in an undersaturated zone, and then the process of equilibrating with the reservoir takes us into the nucleation zone and then into the metastable zone for crystal growth. So it's important to realize that these two setups are sort of operating along a different pathway to achieve crystallization. With microbatch under oil, you have very minor dehydration over time, but you can also modulate this evaporation rate with different oils. So specifically, we use paraffin oil, which is very, very resistant to dehydration, but oils like silicon oil are a little bit more permeable to water, and then Al's oil is sort of a mixture of different oils. So all of these things have sort of been used in different success rates with microbatch under oil crystallization. The other thing I wanted to mention is scoring crystals. And so this is the Hampton Research based scoring protocol here. And so we score from 0 to 9 based on effectively how productive our crystallization experiment has been and how likely it is that the results of our crystallization experiment will lead to something that we can actually collect crystal-based diffraction data on. And so, of course, we start with the undersaturated clear condition. Phase separation, this is something we often see, especially with something like a membrane protein. Heavy precipitate, so this indicates that we've sort of way overshot our nucleation phase and moved into precipitation. Light precipitate indicates that we're sort of getting closer to our nucleation concentration, but we've still overshot it by some degree. I've definitely seen both heavy and light precipitate clear up into crystals over time. So these are things that you might need to monitor over the course of time. For our microcrystals or birefringent precipitate, these are usually sort of intractable by any X-ray based methods. Although I think we're sort of working towards a point, especially at beamlines at Diamond, like VMXI, where you can actually use microcrystals in situ for diffraction. Urchins, spheroids, and dendrites. So I'm sure we've all seen things like this. And so this is protein crystallization that we're seeing. It's just unfortunately in a form that's really hard to collect data from. The same thing from needles. They're very thin. And so it's extremely challenging to get the intensity of diffraction that's needed, as well as, of course, needles will be very anisotropic as well. The same thing with plates and 2D crystals. Again, very challenging to handle, and they produce less than ideal data. 3D crystals that are less than a micrometer. This is a situation that we would be very happy to sort of see from a screening experiment. We can use these as seeds to optimize. You can modify or modulate the grid screen to try and improve the size of these if you're interested in doing single crystal experiments. But then, of course, our bread and butter, our favorite thing to see in a crystallization plate is going to be 3D crystals. If they're greater than 200 microns, you're almost guaranteed to be able to collect a full data set from them if they diffract. So this is just a good way to kind of get a sense of the direction that your screening and your grid optimization is going in. If you're starting to see more and more of these 6, 7, 8, 9s in your plates, you're really having a sort of productive optimization. Whereas if you're sort of plateauing in the 0 to 4 range, you may want to consider moving on to a different sort of condition to optimize. So now I'm just going to talk a little bit about how we get our crystals in front of the beam. And And specifically, I just wanted to make us a little bit familiar with what the setup for diffraction experiment, especially at a synchrotron, looks like. And so you have your X-ray beam, and then we need to put our crystallized molecule in front of this beam. And then you will see the diffracted rays on some sort of detector at your beam line. And so what do we want from some sort of mount where we can mount our crystals in front of this X-ray beam? We want it to be stable. If it's not stable, we're not actually able to get data frames that correlate well to one another. And so it'll be really challenging to handle these data. We also want it to be able to rotate. This is important so that we can collect data in all 3 dimensions of our crystal lattice. It needs to accommodate different shapes and sizes of crystals. So for example, you might have a cubic crystal, great, that's wonderful, but what if you have a plate? What if you have a needle? What if you have, like we had recently, like a 2 micrometer long crystal? This is important, and these data sets or these crystals can also yield data sets, but you need to be able to use them. We want to be able to stabilize the crystal with some sort of mother liquor, but mother liquor also causes background, so we don't necessarily want too much mother liquor. So the solution for this that was sort of devised are using crystal loops. And so the size range tends to go from about 0.025 to 1 millimeter. And what they look like, they have a base, typically they'll have a microtube, and then you have your tiny loop where you mount your crystal at the end. The base of the pin is magnetic so that you can use tools to manipulate these and also put them in pucks, which are magnetic as well, and stick them on goniometers, which are also magnetic. Usually crystal loops are composed of X-ray transparent material or nylon that has very low background so that it doesn't interfere with the diffraction that you're seeing on the beamline. So this is a nylon loop that you're seeing right here with a little red crystal sort of suspended in the mother liquor here. Mitogen loops look a little bit different. So they are composed of this X-ray transparent material. And then you can see you have your loop on the end with a little crystal there. And this is just kind of a profile of what these pins look like with the little loop on the end. And they come in these boxes so that you can put them into different bases depending on your needs and the needs of your beamline. You can loop your crystals directly in the stabilizing solution. And so for a long time, this was sort of the thing that held back cryocrystallography. So since the 70s, we sort of understood that crystals could withstand a lot more radiation if they were cryoprotected or cooled to about -75 degrees Celsius. But it wasn't until Elspeth Garman started looping in mother liquor and cryoprotectant that we were actually able to take good advantage of this. So other mounts and strategies for single crystals, it's not just loops. We also have micro meshes. These are especially useful for, we've used them in the past for microcrystals. So if you have some sort of slurry and your crystals are and you're sending your crystals to a microfocus beamline, you can sort of loop them and maybe you'll get a couple like dotted around on your micro mesh. And then you can collect multiple data sets from the same mount. An angled tip mount can be useful if you have a crystal that's really long in the C dimension. And so this sort of gives you a little bit of an angle so that you can access the 3rd dimension of the crystal and you're not just rotating around it when you're rotating your goniometer. UV vis mounts, these are especially useful if you are trying to collect some sort of UV vis data. Like Sarah was talking about earlier, for example, this might be to assess the oxidation state of a metal center. Micro grippers, these are kind of interesting. I've never personally used them, but they're kind of good for if you want to loop a needle crystal with sort of this shape or plates. I think you can loop really well with this sort of micro gripper. And of course you have reduced background here because you're not sort of looping in that big pool of mother liquor as well. Quartz capillaries can be quite useful. They're UV invisible or UV transparent, so they're quite good for the same application, UV vis experiments. There's different chips and even like microfluidics devices that are now being used, especially at XFELS. And then, of course, I mentioned the in situ plates. So these crystals are actually being experimented on. There's data, X-ray data being collected on these crystals while they're still in the crystallization plate that they were grown in. And this is at Diamond Light Source, VMXI. So I did want to mention quickly goniometer bases and automounters. So each beamline sort of has their own setup and robots for how your pins are mounted onto the goniometer for X-ray data collection. And Mitogen has a fantastic beamline guideline that tells you exactly what sort of pucks and pins are accepted at your beamline. So I highly recommend if you have an experiment set up and you aren't sure what to do, go to this website and they'll be able to tell you. But the most common bases are these spine pins, and these are actually hollow in this region. And then the ALS pins, they have this sort of conical shape, and so they are not hollow. So it's really important to kind of know the difference, because if you send spine pins to SSRL, they will just send them right back to you and they will not be able to do your experiment. So pucks and dewars, unipucks, this is kind of the universal puck. And so a lot of people will use these to ship their samples. They have a magnetic top and a pin that sort of inserts into the bottom, and they have 16 spaces for your crystals. I'm sure Sylvia will talk a little bit more about this, but this is the SSRL cassette, or sometimes people call it the pineapple because it looks a bit like a pineapple. But it has essentially the same function as a unipuck, but you can fit quite a few more samples in here. Samples are typically transported using these dry shippers. So the way these function is that you load up this material that absorbs liquid nitrogen in your dry shipper a few days ahead of sending it. And it will actually, if your dry shipper is functioning properly, it should keep your samples cool for about a week. And so this is typically the way that frozen crystals are transported to synchrotrons and back. Another thing I wanted to mention, like we sort of talked about before, salt crystals can be a huge complication. And I'm pretty sure every crystallographer has accidentally looped a salt crystal thinking that it was protein and was sorely disappointed to see the diffraction on the beamline. There are a few tells. The first is that protein crystals will crush like a soft cheese. I like to say like a medium brie, due to a high solvent content. So the solvent content can be anywhere from like 20 to 60% in protein crystals. So highly composed of solvent. They absorb dyes like crystal dye, which I think is sold by Hampton, into the solvent channels. They're kind of weakly birefringent, so that's what we're seeing here. And you can observe really rapid dehydration damage when you remove it from the drop. And you might even see this when you just open your drop and it's sort of exposed to the environment, is that your crystals will start cracking. And the reason why is because of the high solvent content. These crystals should also have a UV signal. Salt crystals, a lot of times you can just tell visually what are salt and what are not, because it's very likely that salt crystals will exhibit a bunch of different morphologies. It's possible for protein crystals as well, but I think it's more common to see that in salt crystals. They clink and they sort of fragment when crushed instead of sort of crushing like a soft cheese. They're hard. They exclude dyes because they don't have the same extensive solvent channels as protein crystals. They're strongly birefringent, so you can see them sort of glowing here. You don't observe a change in appearance when removed from the drop. You have your container of table salt, right, that you season your food with. It looks the same as if it just came out of solution, and that's because it doesn't have these solvent channels. You don't observe a UV signal. And that is a bunch of different ways that you can figure out what's protein and what's salt before you are sorely disappointed at the beamline. So next, I kind of want to talk about something that crystallographers should be thinking quite a bit about during their experiment, and that is mitigating radiation damage. So X-ray data collection experiments are really a balance between collecting good, high quality, high resolution diffraction against mitigating damage caused by the beam. So whenever you hit your crystal with an X-ray beam, it is going to cause damage to your crystal. And so this is actually just sort of a very striking example of X-ray radiation damage on a protein crystal. And so what they're doing is they're moving the beam or moving the crystal and exposing to about 0.5 seconds of the beam. And you can see this crystal is slowly being destroyed by the exposure to the beam. So it's not always this dramatic, but it is still likely happening to your crystal, whether you can see it or not. And so we like to divide radiation damage into sort of two categories. We have local damage and global damage. Local damage is the sort of thing that's going to happen at lower doses of radiation. So here I have about 4 megagrays of dose. And the incident X-rays actually cause the expulsion of lower energy electrons and free radicals that cause damage to specific sites on your protein that are electronically sensitive. And so specifically what I'm talking about here is you can see residue decarboxylation. Any disulfides you see, you might see a little bit of damage there. And especially you're going to see photo reduction of metal centers. And so if you're really interested in figuring out what your protein looks while it has a reduced metal in a metal center, this is going to be really challenging to do because you're going to start seeing photo reduction really rapidly with a lot of metal binding proteins. Global damage is another thing that you will experience, and this is just the phenomenon of being, exposed to the X-ray, and the ultimate result is you're going to experience a global non-isomorphism, and so your individual unit cells are going to no longer be sort of experiencing long-term symmetry anymore. And so this is going to affect your high resolution diffraction. You will probably see an expansion in your unit cell, increased mosaicity. So you might see your spots smearing or getting elongated. And if you're able to solve your structure, you'll sort of have a global B factor increase. So this is a temperature factor or an atomic displacement factor that I'll talk about a little bit more. So on the left here is a graph. And so the relative B factor, temperature factor, essentially what this is telling you is how much your data reflects that specific atoms are moving in position. Versus the dose in megagrays that these protein crystals are receiving. So we have lysozyme, catalase, and diamonds, thaumatin, and apoferritin. And what you can see here is that across the board, as we increase the dose that these crystals are receiving, the B factor is increasing. And so we can sort of use this, at least in this case, as a proxy for radiation damage that's being experienced by these crystals. Another interesting thing to sort of note about this chart is that it's obvious that these crystals are experiencing this radiation damage at different rates. And so it is going to sort of be sample dependent, the rate at which your sample is receiving radiation damage, and these B factors are increasing. So this is what I was talking about in the 70s, where Haas and Rossmann actually discovered that cooling a crystal down to -75 degrees Celsius could actually improve its lifetime. And so we're looking at the signal to noise ratio versus the exposure time. And we can see for the crystals exposed at 25 degrees, there's an extremely rapid decay in the quality of the data as it's exposed to the X-ray beam, whereas the cooled crystals have quite a longer lifetime and they have a slower rate of decay. And so this could be really beneficial for us if we learned how to harness this and appropriately cool crystals without destroying them. And that's exactly what Elspeth Garman did in the 90s, where she perfected the technique of cryocooling macromolecular crystals in the presence of their own mother liquor and the cryoprotectant, which sort of keeps them hydrated and protects them from the effects of being rapidly plunged into liquid nitrogen. So in fact, this is really exciting for us because at room temperature, it's sort of been determined that the average crystal can endure about 0.1 megagrays of exposure, whereas a cryocooled crystal can endure about 20 to 150 times more exposure than these room temperature crystals. And so this allows us the ability to sort of collect more intensity of data as well as more frames of data from the same crystal, so we can get full data sets from single crystals. So cryocooling, pros and cons. Of course, there are pros and there are also cons for this technique. It can't all be rainbows and butterflies. So we can have improved crystal lifetime. So again, we can collect that full data set from a single crystal, which is really powerful. We can use more intense data collection parameters to sort of increase the intensity of our diffraction. This sometimes can also boost resolution by several angstroms. And the reason why is that sometimes cryoprotectants can influence how tightly packed our unit cells are. And oh, I'm getting fireworks. But it can also sometimes do the opposite. There's also more reliable shipping and transportation options for cryocooled crystals. It's a little bit challenging to keep room temperature crystals in a hydrated environment so that they'll still be able to diffract on the beam. There are some options. There's an SSRL shipping box that's been used for room temperature crystals, but by and large, it is far easier to ship frozen crystals than room temperature. Some cons. Cryocooled crystals will almost always have increased mosaicity compared to room temperature. So that's something you're going to have to work around. And sometimes it's useful to have both a cryocooled and a room temperature data set, if that's possible. It requires quite a bit of optimization to determine the best cryoprotectant. And sometimes this can really be heartbreaking when you only have one perfect crystal to send and you dissolve it in cryoprotectant. Also, freezing might stabilize non-physiological states or immobilize mobile residues. And so this makes it challenging to sort of interpret our structures and analyze B factors to determine where these proteins are sort of experiencing dynamicity because it's artificially constrained by having been cryocooled and frozen. Cryocooling also uncouples the global and local radiation damage that we're seeing. And so let me go back. Here I was talking about local and global damage. For room temperature crystals, these are effectively occurring at the same time. But in cryocooled crystals, we definitely see a difference between when local damage is happening at this lower exposure and global damage happening at this higher exposure. So this is something to keep in mind as well. So what is the goal of cryocooling? We have to cool these crystals so quickly that they're encased in vitreous ice. And the reason why is because crystalline ice diffracts on the beam and it will produce these rings of ice that make it a little bit challenging for our data analysis programs to deal with our data sets and obscure diffraction from our protein in these specific regions of the frames, the data frames. So this glassy appearance that I mentioned of the loop, so it looks like this, you can see it looks quite clear in the middle. It's necessary, but not sufficient for good cryoprotection. So this is our ideal case here. There is a little bit of ice on the outside of the loop here, but it is not going to affect our ability to collect data from the protein crystal that's in this vitreous ice. This is our ideal situation. This crystal you can see is a little pizza slice here. It does have some ice contamination on the surface, so we might experience some ice rings in certain frames of this data. And then something that everyone who has frozen crystals for the first time has experienced is the snowball. This means that your crystal is highly contaminated with ice. Sometimes you can ameliorate this by annealing your crystal on the beam or popping it back in the dewar and putting it back on the goniometer. But to achieve vitreous ice, the loop must be rapidly plunged into liquid nitrogen, and you just want to make sure you're avoiding ice contamination on any of your instruments and in your dewars. That's the best way to keep your crystals ice-free. Okay, so cryoprotectant solutions, Justin had asked about this a little bit. This is a figure from one of Elspeth Garman's reviews, I believe. And here is a series of different cryoprotectants that you're seeing on the right here. But by and large, glycerol is the most commonly reported. It accounts for about half of the PDB entries were cryoprotected with glycerol. Other things, you have ethylene glycol, MPD, oils, different PEGs, alcohols, and then cryosalts as well. Are kind of used in, roughly the same ratios. I would say glycerol is always a good starting point. It works in about 2/3 of cases, and you want to have your glycerol concentration in your final cryoprotection solution somewhere from 15 to 25%. And you might have to freeze crystals that are like 15%, 20%, 25%, 30%, 35% to see which of these gives your crystal the appropriate cryoprotection. You need to sort of consider your mother liquor because sometimes there's already a cryoprotectant in the solution that your crystal was grown in. If you grew your crystal in a PEG concentration greater than about 30%, it's usually sufficient to freeze it without cryoprotection, so you don't need to add anything additional. If it's less than 25%, a good strategy is to just increase the concentration of that same PEG it was crystallized in to achieve that 25 to 30% PEG concentration. And so that way you're not adding anything that's not already in your crystallization condition. Sometimes that's a little bit more delicate. Some salts are suitable for cryoprotection, especially like lithium salts and malonate. I typically don't recommend using a cryosalt unless your crystallization condition already contains these salts, so like a lithium salt or a malonate salt. Other people might have a different opinion. I've just never had good luck with them. But screening through cryoprotectants is worthwhile, and Hampton makes a kit called CryoPro that I find extremely useful if I'm struggling to succeed with the common cryoprotectants. And we found a few new ones that are kind of our new favorites using this. So it's definitely worthwhile. If you have enough crystals, send a puck with different cryoprotection strategies because you might be surprised what things work well for cryoprotection and what don't. So what I like to do is freeze crystals in duplicate with the same cryoprotection. So I'll freeze 2 with like 25% glycerol. I'll freeze 2 cryoprotected with like hexane diol. I'll freeze 2 cryoprotected with some sort of sugar, and so on and so forth. And this almost guarantees that I'm going to be able to find something that's a suitable cryoprotectant without having to go back to the beamline several times. So best practices, these are just, from someone who's frozen hundreds of crystals and made all these mistakes before. You want to plan ahead. So you need loops, liquid nitrogen, a puck or a storage cane, cryoprotectants, tools. You need razors for cutting plate seals. You need wands, definitely notebooks so you can take notes. Sometimes I even like to voice record myself when I'm freezing crystals if I don't like have a spare hand to write things down, and then I can just listen to my recording and figure out exactly what I was doing. You can consider preparing appropriate cryoprotectant mixtures prior to freezing for complex soaks. And so what I mean by that is a lot of times what I like to do is just mix my cryoprotectant on site while I'm freezing. So I'll just pull some mother liquor from the from the cocktail well and add whatever concentration of cryoprotectant I need. But sometimes you will want to have the exact concentration of your mother liquor in your final cryoprotectant solution. So what you actually need to do is increase the starting concentration of the mother liquor components so that when you dilute it with the cryoprotectant, you're achieving the appropriate final concentration. You want to use clean liquid nitrogen. Ice is the enemy here. Work quickly. Dried crystals often lose diffraction potential or get salty. Again, that's because of those solvent channels. I like to try and fit the loop to the crystal as best you can. And part of the reason why is because you want to be able to find it when you're on the beam. And if you don't have the ability to raster, it's going to be a little bit challenging. Crystals that are too big for the loop sometimes are not hydrated enough. Sometimes the circumstance is totally fine, but you do want to make sure that you're keeping your crystals hydrated. You want to use surface tension to your advantage to suspend this crystal inside the loop. And sometimes if you loop it while you're touching the edge of the crystal, it'll actually slide down into the center of the loop because of the surface tension generated. You want to minimize the bubble of mother liquor. And so usually solutions that are really viscous, like high PEG concentrations, will form these bubbles where the surface tension is a little bit higher. So they're sticking off the top and bottom surface of the loop. And so it kind of looks like a circle of mother liquor. This is going to be challenging because it'll produce background on the beam. It's also going to make it more challenging to find your crystal in this bubble unless you have the capability to raster. In rare cases, crystal regions outside the loop outperform those inside the loop, partially because you don't have the background from the mother liquor. I always say that you should just loop a few weird ones if you want. It's like when you're taking photos and they say, let's do a weird one. You should do exactly that while you're cryoprotecting and freezing. Other tips and tricks, it's going to be very sample specific. So just because something worked for something else you worked on, it won't always work again. So keep this in mind. Combinations of cryoprotectants might be beneficial if you're not finding a single cryoprotectant that does work. One way to figure out if your cryoprotectant is at the appropriate concentration is to freeze empty loops of mother liquor and sort of look at them under the scope really quickly to see if you're producing that glassy appearance. Again, this isn't necessarily telling you that this is going to be the best cryoprotectant because you haven't seen the data yet, but at least you're achieving that vitreous ice. Stepwise soaks of cryoprotectant may be useful for delicate crystals. So if you pop your crystal into 25% glycerol and it completely dissolves, I recommend maybe starting at 10% and then slowly working your way up to 25%. Sometimes you can do this and maybe you only get to like 20%. Just freeze out of the 20% when you start seeing damage to your crystal. If you cannot diffract cryoprotected crystals, try diffracting them at room temperature because cryoprotection can cause damage to crystals, mosaicity, artifacts. It's definitely worth trying to take advantage of a crystal and see if it diffracts at room temperature or if the cryoprotectant is the problem. If you see damage during a cryoprotectant soak, I've seen this in the literature, never tried it myself, but maybe you will. Leave the cryoprotectant solution on the crystal for 4 to 24 hours to see if your crystals might recover. And the final thing is annealing on the beam can reduce your ice contamination and potentially reduce mosaicity in poorly cooled crystals. So just because your snapshots are not showing exactly what you were hoping for, you can certainly try annealing to see if that improves your situation. All right, and so that's everything I have. So I'd be happy to take any questions.
All right. We got to thank you. So, Okay, you've got a question, Suhas? I'm sorry if I mispronounced it.
No, that's correct. Thank you. Thanks for the talk, Gabby. So can you use plates to, like my protein forms very good plates without much effort. Can you use plates to collect data? Like they're basically 2D, they're not 3D. And I think we tried it once on a loop and it didn't work out.
It did or it didn't?
It did not. So, but we
It
use a
didn't.
normal loop. Yeah.
Okay.
So is there any other way? And yeah.
So yes, you definitely can collect data from plates. The major issue is that one dimension of the plates is typically it kind of hasn't grown enough, and so it's grown anisotropically, and so that's going to be potentially an issue, and it can be an issue for diffraction as well, right? Because you don't have as much information as you need in one dimension. It's tough. If you've already tried it on the beam and it didn't diffract well, unfortunately, I think it's quite challenging to optimize using a grid screen from plates, but I think your best bet is potentially to seed, so to fragment your plates and try to grow a more 3D form if that's possible.
Yeah, I agree. Try seeding them. Break them up and see what happens.
I mean, they form just when I'm concentrating the protein without
Oh, wow.
really,
Okay.
yeah, but they don't diffract. So yeah, so I'll see if I can seed the plates while setting crystals or something. Yeah.
Sounds good. If your protein's crystallizing like that, just from concentrating too, it's possible that I don't know how much screening you've done for like different crystallization conditions, but it might be worthwhile.
I had sent my samples to your
Oh, you
facility.
sent it to us.
Yeah,
Okay.
yeah.
Great. Then you're doing
Uh,
exactly the right thing.
Yeah.
All right, so I think in the interest of time, so that was a great presentation, Gabby. Thank you so much. I learned stuff, which is awesome. I think in the interest of time, we're gonna have Sylvia kind of take over. So Sylvia Rusi from SSRL, and she is going to give us kind of a general talk about shipping samples. And then also some specifics about shipping to SSRL. And then we're also going to hear from NSLS2 and Chess about shipping samples and kind of beamline specific things. So take it away, Sylvia.
Yeah, I'm gonna start actually discussing a little bit. Can you hear me? Can you see the screen? Everything looks fine?
Everything looks good.
Perfect. So I'm gonna before going into the tips and tricks about sending samples to SSRL, I like to give you like a short overview about the structural molecular biology, beamlines, and what we can offer to you and how you become actually an SSRL user. So the structural molecular biology group at the Stanford Synchrotron Radiation Light Source, for those that doesn't know it, runs and operates not only the macromolecular crystallography beamlines for X-ray diffraction, but also the small angle scattering beamline and the X-ray spectroscopy and imaging beamlines. In this slide, you can see the SSRL map and how those beamlines are located around the storage ring. You can see in red beamline 9.2, 12.1, and 12.2, and 14.1. Those are our beamlines for X-ray diffraction, but we also have a beamline 4.2 for small angle scattering and a different number of beamlines for X-ray absorption or X-ray imaging. I'm going to specifically talk about the macromolecular crystallography beamlines, but if you are interested in using or getting beamtime in any of the others, I would advise to contact Aina Cohen, and I will give you later, you will see her email address in other slides. So because she can help you and point you to the person to ask about potential beam time there. But coming back to our beamlines, the X-ray diffraction beamlines, this is our web page here. And if you click there, you will have a table with a description similar to the one that you are seeing right now, with a description of all our beamlines, beamline 9.2, 12.1, 12.2, and 14.1, as I mentioned before. All of them are tunable. They have a really wide energy range from lower, even lower as 6 kilo electron volts up to 18 kilo electron volts in the case of beamline 12.1. Different fluxes, ondulator beamlines, 12.1 and 2 are the most intense beamlines that we have. And in some of them, we have very special instrumentation that makes those beamlines unique. Regarding the detectors, we have also different beamline sizes, microfocus beamline, another for if you have samples a little bit larger. And regarding the detectors, in 12.1 and 12.2, we have Eiger's detectors, while on 9.2 and 14.1, we have Pilatus 6M detectors. However, during this shutdown, we are planning to upgrade the detector on beamline 9.2 and have an Eiger 16M there too. Let me highlight a little bit the capability of some of our beamlines that really makes them unique. As Sarah mentioned, we have a online UV visible absorption microspectrophotometer at beamline 9.2. This is extremely useful for people that works with metal enzymes and is trying to assess the oxidation state of the metal and see any potential impact of the X-ray exposure during the data collection. Actually, we can interleave the microspec snapshot, like you see here, with the diffraction collection to monitor any X-ray induced structural changes of the redox center. We can, in general, confirm the identity of reactions intermediates that they are trapped within a crystal and directly link a structure to its position along the reaction pathway, or we can monitor reactions that are happening in the crystal. So you can trigger reactions and monitor them. There are a variety of applications for UV visible absorption spectroscopy, and the beauty of this is that it's fully remote accessible. So you don't have to be at the beamline or do some difficult mounting of preparation of your experiment. You just ship your samples to beamline 9.2. We enable you at the beamline, you mount samples and start working, either collecting the spectra or the X-ray diffraction. If you want to know more, you will see that in all of my slides at the bottom, I have the link to our webpage with the specific information of this particular application. Of course, we don't have to forget about the state-of-the-art microfocus beamlines, 12.1 and 12.2. On the left of your screen, you have beamline 12.1. On the right is beamline 12.2. They are both microfocus. We really focus the beam very narrow mainly in the vertical. So for beamline 12.1, we have 5 micron vertical beam, focus beam, and 15 microns for beamline 12.2, while the horizontal is a little bit larger, is 40 in both beamlines. On beamline 12.2, we also have what we call the microcollimator, so you can further reduce the size of the beam to 15 by 15 microns. So those beamlines run in an ondulator, and they are a high flux beamline at 12,658 kilo electron volts. This is 410 to the 12 photons per second. And as I mentioned before, they both have the latest Eiger 2 XE16M detector. Beamline 12.1, in addition, is the place where we set up all our remote access program for room temperature. So I don't know if you are aware, but about 3 years ago, SSRL started offering remote access to room temperature, high temperature, and also control dehydration experiment. And everything happens here in Beamline 12.1. So this is actually me loading samples for room temperature data collection in these trays that I'm going to describe later. And these trays, you see, you can ship your samples very safely, and you will learn how in a little bit. And collect data fully remote, because basically what's going to happen is the robot will take care of mounting the samples in the same way that would do for the cryo samples that you ship to the Beamline. We plan to have also this setup on Beamline 9.2, the one that have the microspec. So you might combine or do not only measurements at with cryo samples, but potentially you could do this also remotely and at room temperature. And last but not least, I wanted to mention to you that all our beamlines allow to do what we call an excitation scan, which is extremely useful tool to identify the metals in your samples. So the excitation scan is done with a fluorescence detector, same fluorescence detector that you use for doing the MAD, the traditional MAD scan when you have a derivative. Okay, so this fluorescence detector will record or will measure the fluorescence count from any element that is present in the sample, and which has any absorption edge below the excitation energy. So if this is the excitation energy, anything that has a fluorescence below that, it will show in the scan. And as you can see in this scan, the person that excite the sample discovered that had on top of selenium, they have zinc on it. Actually, they have, the peak is proportional of the amount of the metal that you have. And yes, you have more zinc than selenium actually in this sample. Again, all information about how this works is here and is available in any of our beamlines. So how you get beam time at SSRL? Is pretty easy and it's pretty straightforward. So you need only to submit a SSRL macromolecular crystallography proposal. And there are 3 different kinds or types of proposal. The standard one, rapid access, or the block allocation group proposal. A standard one is the general proposal. They become, once they are approved, they become active for 2 years, and they can be extended for an additional 2 years. Rapid access is in the case that you are really in a rush and you need to get beam time yesterday. So in that case, you can submit a proposal, and we are going to do our best to give you beam time as soon as possible, and always within a month from the time you submit the application. Of course, this kind of rapid access is not meant for long-term use, but might be useful and might be a fast way to get beam time if you truly need it. And of course, for a group of researchers that want to combine short beam times and do like a macro proposal, we have the block allocation group or the back proposals. And as you can see, no matter which proposal you want to apply to, all the information is here in this web page, and you can go there and read the very, very careful prepared guidelines to how to apply. So in general, I would say to submit a proposal, what you do is you have to use our user portal. This is the link to it. And there are 3 submission deadlines per year: December 1st, April 1st, and July 1st. But I would say we are super flexible. Don't worry if you miss the deadline and you still want to submit a proposal. I would say talk to or contact Lisa and Aina, and they will make sure that they will help you with the process, and if there is any possibility to give you the beam time, we will do it. Yeah. So yes, and even if you are not sure if you want to be a necessary user and you are, well, maybe I would like to try, but I'm not sure. Okay, send an email to Lisa and Aina, because we can coordinate with you. We sometimes offer even if you are not formally a user, we offer some few hours of beam time, and we can have like sessions where we show you the beamline and what is possible to do there, and what kind of results you can, or what you can expect from our beamlines. Okay, and in our webpage we have a lot of information. Yeah, and if you go to, this is a link, and if you go to the user guide, you have an extensive number of information there. Since from the beginning, how you become an SSRL user. Also, this will give you the link to the how to prepare the proposal, how the proposal have to be formatted, etc., etc. And you can read this, but again, if you think it's too complicated, don't worry. Send an email to Lisa and Aina or me, and we will guide you and will help you during the process. And with this is my first part of the talk, and I don't know if someone has, I see four questions maybe in the chat, because maybe we can stop here. I can answer the questions if are related to this, and then I can continue with the second part of the talk, which is more specific oriented to preparing the samples and some tips and tricks.
Sylvia, I don't know if there's any specific.
Oh, okay. Sorry.
I think there's
Yeah.
a kind of follow-up.
My chat was not was okay. Perfect.
So I think
my question then is, do you have any question that you want me to answer now, or I can keep going to the second part of my talk, and at the very end I can answer all the questions. It's up to the audience.
Yeah, let's go to the second part.
Yeah.
and
we keep going then.
Yeah.
Okay. Okay, so changing subject, and now I'm gonna focus in some tips and tricks about sending samples to SSRL. Okay, I divided the presentation in 2 parts. I'm gonna first talk about the cryogenic samples, and in the second half about the room temperature samples. And when we think about cryogenic sample, this guy here, the cryogenic chip endure, is our best ally. This is our best friend, and we have to treat it carefully, because our samples depend on how well we keep our chip endures. This is why I decided to start, before telling you how you mount samples, what are the sample containers, etc., give you some guidelines on how to test, prepare, and ship your dewar, because this is extremely important. So our samples truly depend on that we do a good work with it. And everybody wants to avoid the sample loss during shipment, obviously. And to do that, it's very important to at least verify the shipping dewars every 6 months. And in an ideal world, it would be every each use. I know that might be too much sometimes, but at the very least, twice per year, you need to do several tests on your shipping dewar to make sure that it's in good shape. Why? Because a fully functional dry shipper can keep samples frozen for 14 days. That's quite a long time, and sometimes it's needed because FedEx or UPS, whatever is the carrier that you are using for shipping samples to the synchrotron, Sometimes they experience delays on the shipping and the samples don't arrive or whatever is the situation. So you need to make sure and have the peace of mind that the samples are going to be safe during and kept at cryo-cool during the transportation. And unfortunately, those viewers don't live forever. So at some point, they start showing some performance degradation. And the main reason of that is could be vacuum loss. Usually it's a gradual vacuum loss, but in very bad cases, we have what we call the catastrophic vacuum loss. And once the vacuum is lost, the samples will arrive at room temperature, and the whole viewer will arrive at room temperature to the synchrotron place. This is what we really need to avoid. There's another problem is, with the use of the viewer, you can have accumulation of moisture in the absorbent material that you have inside this viewer, and this the material that absorb the liquid nitrogen. And of course, depending on how well or bad you treat your viewer, you can damage or lose the absorbent material. So let's see what we can do to check our viewer. But before that, we need to start thinking how we have to charge our viewer, because sometimes what happens is not that the viewer is faulty, but the people didn't charge properly or didn't prepare properly the viewer before sending it to the synchrotron. To properly charge the viewer, what we have to do is you pour liquid nitrogen until it's full. Yeah, and I have to say that please avoid any spillage of liquid nitrogen on this guy here. This is the vacuum plug. And if you're wondering what is this piece of foam doing there, so to our users, if you send viewer to SSRL, we are making and putting these protective caps on the vacuum plug, and we ship your viewer back with it. So because it's extremely important, to protect it. Okay, so you pour the liquid nitrogen until it is full, and then you put the cap on the viewer. You wait for 20 minutes and repeat the process again until the nitrogen, the liquid nitrogen remains always at the top edge of the viewer. That means when it's not going down, it's always there. It means that all the nitrogen that the material was able to absorb was already absorbed and is fully saturated with liquid nitrogen. And then we are ready to pour out the excess of liquid nitrogen, making sure that it is fully charged and ready for shipping. That's the proper way that we should always charge our shipping viewers. What about the test that we can do at least twice per year to check if they are in good shape? Okay, there are three main tests: the vacuum failure test, the absorbent material, and the viewer insulation test. The vacuum failure test is super straightforward. So you charge the viewer following the procedure that described earlier and remove all the excess of liquid nitrogen. So you wipe the outside of the viewer, because sometimes, it's like a little wet or has some condensation because of the filling process. And you wait 2 or 3 hours. And if after 2 or 3 hours you see any excess or condensation or frost on the outside of the viewer, that means the viewer didn't pass the vacuum test. And therefore, you should seriously consider either repair or replace the viewer. I actually don't know if it is possible to repair cryogenic viewers. I would say most likely you will need to replace it. How you test the absorbent material? Okay, now we have the viewer at room temperature, and it is empty. So, and then you weight it, then you charge it and weight it again. And if you subtract the cold weight minus the warm weight, the amount that it will give you is the amount of absorbed liquid nitrogen in the viewer. Depending on what kind of shipping dewar you have, this should be 3.6 kilograms for the Taylor Wharton dewars or 3.1 kilograms for the MBE dewars. So this is to give you an idea. And what about the dewar insulation? How we test that? Okay. So again, we start charging the dewar, remove the excess of liquid, and we let it sit an hour or so, and we wait the dewar. And then we repeat the same measurement 24 hours later. Obviously, the weight will be lower 24 hours later because there will be some mass lost. And you calculate the mass loss in between the measurements. And what you have to make sure is that mass loss is about or not more than 150, 200 grams per day. Yeah, this is for a newer model. Of course, if you when the dewar gets older, you will have a bigger mass loss. But again, this is a good reference for you to know if it is, to give you an idea how bad or good is is keeping and is the dewar insulation. All the tests that I mentioned, again, you have them here in this in the below link, and you can try to do them because it's extremely useful. Okay, our dewar is ready. We can start thinking about harvesting our samples, and Gabby gave super nice tips about how to do that in the best way possible. But as she mentioned before doing any harvesting, you need to think about what you are going to do what you are going to use for harvesting your samples, and what kind of sample containers are you going to use. Because depending on which synchrotron or beamline you are sending your samples, it could happen that some kind of sample containers are not compatible with the robot there, and the same with the sample pin bases. And as you can see in this picture, there is a variety of them, and I'm pretty sure that I may be missing something. But it's normal to be confused, yeah, especially if you collect in different synchrotron size. Why is happening here? Why if in a place this loop works, but in your place doesn't? Okay, so what you have to do is always ask the user support person. You can go to the web page that Gabby showed you. I didn't know that even exists, but I have our own. We have our own page. Is the robotic sample handling page. This is quite old. Started like probably, I don't know, more than 15 years ago, and we are keeping, Anna and I are trying to keep this web page up to date, and we ask typically, other synchrotrons beamline scientists to give us information. But in this web page, you have all the United States synchrotrons, Canada synchrotron light source, but also is worldwide. So it's the worldwide list of the synchrotron sites. And if you select your synchrotron, it will give you the beamline name, which is the kind of robotic mounting system, which containers are supported, and which kind of pins are you can send them for collection. And here is the link of this page. Specifically talking about the SSRL, so our SAM robot allowed to mount two different kind of sample holders. The SSRL cassette, I never knew that it was called Pineapple. That was that was a first for me. Okay, and then we have Unipacks. So either SSRL cassette or Unipacks are compatible with our robot. We have 3 positions in our robot. If you are working with cassettes, we can load up to 3 cassettes at the same time in the robot. If you are working with Unipacks, we have a special adapter where we mount 4 Unipacks per adapter, and we can have 3 of them inside the robot, meaning that you can have up to 12 Unipacks at the same time. And regarding the sample pins, I know spine pins are common standard, especially because it's a European standard. But unfortunately for our robot, they are not good because too much magnetic material was removed. And we ask people sending samples to SSRL to use any other kind of compatible sample mountings, because spine pins give problems with the robot. So we are going to investigate the possibility to change that. So, but we might need to do some modifications at the beamline level and the robot level. So, but we are working on it. We are trying to open the possibility to that you send also spine pins. But for now, we prefer you don't do it. And although many people use Unipacks because are compatible in many different synchrotron beamlines, we have our special cassette, and we offer to all our users an SSRL cassette kit. We have a loan program, and as soon as you remind our user and keep using the cassette, you can keep it. It's completely free for you. You just need to request it. By sending an email to Lisa or Aina, and we are going to send ship the kit that is shown here, where include the cassette, the canister to put the cassette, and the special handle to manipulate the cassette. And I didn't mention, but per dewar you can load up to 2 cassettes. So it's about 200 samples shipping in a single dewar. Okay, few tips about the preparation of the pins. Why the pins are so important? One of the very important things is to check the dimension of the loops. In our beamlines, the cryostream is located like horizontally, is on axis with the phi axis. So you might not have much problem if the loop is a little bit larger or shorter. But on other synchrotrons, the problem is the cryostream is pointing 45 degrees towards the sample, and you don't want to mount the sample and the sample is too short or too long that is outside the airstream, the cryostream. So it's very important that you check with your support staff if they have any specific requirement about the dimension of the loop. We do, and everything can be found in our webpage, or even you can look in this video where describe how you prepare the pins for shipping to the SSRL. Of course, you will need some kind of glue to attach the loops to the magnetic base. And for that, we strongly recommend epoxy, not super glue or not other kind of fast drying glues, because Aina have done an extensive work testing many different kind of glues, all of the ones that are shown there. And at the end of the experiment, if you want to really enlarge the lifetime of your loops and also prevent problems with your robot, the epoxy glue was the best option. And please never use an excess of glue, because if you have like a lot of glue around the base of the pin, the magnetic base, that might interfere with the gripper of the robot. And of course, everybody reuse the pins, so there are no disposable. We dispose them, but after many years of use. But if you reuse the pins and you clean them, that's fine. But always be aware to check for any signs of rust or oxidation. and never ever use corroded pin bases. If you have corroded pin bases, just toss, that's it. That toss that loop, that magnetic base, and buy new ones, because I have to tell you, this is real. What I'm gonna tell you, we have a very rusted pin bases on Beamline 92, and the outcome of that was the goniometer head and also the robot gripper were damaged. So we need to replace them both because of the rust got stuck on the goniometer, and then it's like when the gripper went to try to remove the sample from the goniometer, rip out the whole goniometer head with the pin on it. And other very important thing is remember that you are using magnetic bases, because all goniometers have some kind of magnet there. So do not stick anything on the magnetic base, because you especially we have experience with someone that stick these these labels that we use for the Eppendorf. Okay, they put it on the base of the magnetic base, and what happened, they didn't stay there, and they transfer everywhere from the robot goniometer to the magnet. Was really a disaster. I think it's better to use, if you need to color code somehow the pins, use a permanent maker. And I would say try to not I wouldn't do it in the base of the pin, but here on the side. If you need to mark somehow, you can do it on the side. And yes, as I mentioned earlier, here I leave you this link where you have a tutorial, a video that will show you how you prepare the pins for shipping them to the synchrotron, to the SSRL. And last but not least, when we are talking about cryo samples, if you are loading packs, please, please, please never forget the locking rod. There is this locking rod. You see, it's like, why so important this locking rod? It doesn't look like a key element in the pack holder. But they are extremely important because it will prevent the packs for moving or displaced during the transportation. We also have cases where people do not put the locking rod, and when we try to take out, like is shown in this picture, that someone is taking out from inside the dewar the pack holders, we were not able. All the packs were stuck on the dewar, and we could not remove them. So basically, that user lost the beam time because not adding the locking rod. Yeah, because the only way to remove it was to warm up actually the dewar. And when you ship the dewar and everything is ready, remember, do not procrastinate. You can procrastinate with anything else, but not with the shipping of the dewar, because unfortunately, there are delays. Yeah, especially in winter, many times there are delays, and it's better to send the samples, during the first days or the beginning of the week. Why? Because most of the facilities, including the SSRL, they don't work, the shipping receiving department doesn't work or receive any sample over the weekend. And that's totally true for us. So please try to send the samples at the beginning of the week. And if you can do it like 3 working days in advance, it's perfect. So it's always better that the samples arrive earlier than they arrive late. Because if they arrive earlier, we are going to keep them, we are going to refill the dewar with liquid nitrogen, keep them safe. But if they arrive late, unfortunately, we need to reschedule your beam time, because it is time that you completely lose. This is about cryo, but what happened with room temperature samples? Okay, this is a totally different story, because room temperature samples are much more delicate. And I'm gonna specifically talk about the sample temperature samples that you can ship to SSRL. Other synchrotrons may deal with this in a different way. So we decided to automate, as I show you, the room temperature data collection experiments. For that, we designed this special SSRL sample container. Is what we call the SSRL in situ plate. And when you look at the plate, you can identify some common features with a unipack, because you have different ports, and magnetic rings there to load the pins and keep them in place. But also has reservoirs, and you can actually use these trays either to directly grow samples in here and ship to the synchrotron, or you can transfer and load the room temperature samples in the tray. So what kind of sample holders can you load inside this tray? They are shown here in the bottom. Could be regular loops. Of course, magnetic base have to be compatible with some robots, so spine bases are not good, but all the rest are okay. You can use any kind of microfluidic chips. Mitogen also has chips for serial crystallography that are sealed. You can use capillaries, traditional capillaries, or the Mitogen sleeves, like is shown in here. And this is basically the different kind of ways that you can ship your room temperature samples inside that tray. So the tray is compatible with robot mounting and at beamline 12.1, as I show you, but we are going to planning to have it also on beamline 9.2. Let's have a look a little bit more closer look of the tray. How we keep the moisture of the sample during the transportation. And to do that, we have a reservoir, and inside the reservoir, we insert the silicone caps. They are disposable because the idea of this tray, the tray is not disposable. This is like you have to imagine, this is like my pack for my cryo sample. Okay, this is my plate for transport the room temperature samples. So the silicone cap, you can, if you are going to crystallize protein, you can put there directly the mother liquor. But for transportation, we don't want to, the liquid to spill and ruin your sample because, the vibration or the motion during the transport. Because of that, is that we insert inside this silicone cap a tiny piece of foam. This is a special, very absorbent foam, and we soak that foam with 300 microliters of the mother liquor. And you might not see in the picture, but the plate is sealed. There is a tape that we put here to seal the plate and make sure that the moisture that you have in the reservoir preserves the sample that you are going to load there. You can load bare loops, bare crystals in loops, or you can load, as I said, loops that then you put the Mitogen sleeves, capillaries, sealed chips, whatever of the sample holders or mounts that I show in the previous slide. And of course, to make sure that they are compatible and there will be no problem with the robot mounting. So we have to make sure that provide a user some kind of tool, because as you can see here, this is a picture of our gripper, and we have a capillary mounted there. Of course, that capillary is too long. As soon as the gripper tries to close to mount the sample, it will destroy that capillary and your sample will be lost. So that's why on the side of the plate, we have this, what we call the sample size tester. And the idea is very simple. If it fits there, is compatible with our robot, and you will not have any problem mounting that sample at the beamline. And of course, on top of having the sample container, you need to have some thermal shipping container to put inside the trays. We don't send the trays in just styrofoam boxes. That is not good. So we spend time designing a specific thermal shipping container for room temperature samples. These containers are very unique in the sense that once you prepare the container, it will keep the sample temperature, the container temperature up to 7 days, either 20 degree or 5 degree. It depends on how you prepare the container, because you can prepare it for 5 degree shipping of the sample or 20 degree. So how this box managed to do that? Well, the trick is you can see here these phase changing liquid holders. They are covering the sides of the box, but also there is one at the bottom and there is one that we put on top. And to prepare the phase liquid holders, so what you do is you place them 24, 48 hours before you plan to ship at either 5 or 4 degree or 20 degree, depending on the temperature that you want. And then once they are ready, you can make sure that if you put the trays inside this box and you close it, it will keep the temperature. There is also, this rigid polyurethane insulation here around the phase changing liquid holders. And on top of that, we have this thick plastic outer box. We have a shock absorbent foam, because, also it's not only temperature that we have to protect the samples too, it's also some kind of shock or vibrations during the transportation. And inside this box, you can load up to 6 trays, and you can see here we have one of them. And we have these foam inserts, so you can each plate fits in this space, and it will won't allow the tray to be loose or be moving around inside the box. And if you want, you can add, we have a small shell packs that you can add also to stabilize the temperature further. And yeah, so once you prepare your trays and you prepare your box, you can send this to SSRL. And we ask usually people to send it overnight, but the reality is people sometimes doesn't send it overnight and arrive 48 hours later or even later than that. And so far we have very good experience with that. We didn't have any single case where the samples were lost during the transportation. I have to say that if you are going to send samples in crystals in loops, I strongly advise to use the Mightytion mesh loops, because the nylon loops are not good. Most likely if you use nylon loops, the crystal, especially if the crystal is smaller than the loop, they are not good for transportation at room temperature. But the Mightytion mesh loops are really ideal, because it's like a spoon, and the crystal is sitting on top of it, and the mesh will somehow hold the sample. And again, if you want to know more about this, on top of contacting me or Aina, you can go to our webpage and read about the guidelines of how to prepare the the samples for shipping. I have to say that this box can use not only for SSRL because they fit any microplate standard. If you are shipping 96 well plate to the synchrotron to a synchrotron site, you can also use this tray. And with that, and before Sarah kills me, because I run of time. So this is a picture of my group. This is the macromolecular crystallography group at the SSRL that we are there to help you to make your user experience as pleasant as possible and easy as possible. And again, if you have any question, don't hesitate to contact us because we are there for you and because of you. So we will be glad to help you. And now I don't know, I see a number 9 in the chat, but I don't know if that's really 9 questions or there were some other kind of comments.
Yes, there's been a couple of other
Okay.
conversations happening.
Okay.
Sylvia, that was wonderful. Thank you so much. That was very wonderful. And I will say that we have shipped samples to SSRL both using a dewar as well as and room temperature. And we've in fact used that blue box to ship things to NSLS2. And Vivian is about to tell us a little bit about some of the things at NSLS2.
Hi, Sylvia.
Hi, Vivian. How are you?
So in the interest of time, I think we'll hold off on questions until both Vivian and Jenny have gone, and if you're okay for time, for sticking around. But if people have questions, please put them in the chat because we can answer them. I have been answering a couple of questions about metalloprotein things. You can also join in and if you'd like. So our next speaker is Vivian Stojanov from NSLS2, and she will be telling us about the NSLS2 Express, how to kind of get beam time and how to ship samples, and the specifics of NSLS2. So take it away, Vivian. Thank you so much.
Okay. Hi, very nice being here. Thank you, Sarah, for giving us this opportunity. So all the life science at NSLS2 is done by a group that's the Center of Biomolecular Structure. And what I'm going to try to do is to give you a quick tour about what we do, and then talk about how to get access to these beamlines. So our goal at the Center of Biomolecular Structure, and we have partner users, is to have a broad view of biological and environmental problems. So we can go from very small crystals to looking towards some tissues and analyze them. And so we can cover a real large resolution area of or number of sizes, typically from 1 angstrom to about 10 micrometers. And we are constructing a couple of beamlines that will fill in gaps like the one that we can see here. Also, we are covering a large range of energies from about 2.5 keV up to 20 keV. So our center, you can find all about us at this webpage, is concentrated in one of the lab office buildings of NSLS2. This would be the structural biology, we call it Bio Village. Oops. Where we have the MX beamlines. We have 3 macromolecular crystallography beamlines, AMX and FMX, that are they are NSLS2 beamlines, and NYX, which is a partner user program and operated by NYSBC, the New York Structural Biology Center in New York City. And we have then the scattering and imaging program at LIX, which is basically small angle scattering and wide angle scattering, and then an imaging program that just started recently. And we have a partner user program, which is the X-ray footprinting, which is a beamline at operated by Case Western. Nearly opposite to our office, to our LOB5, we have the bioimaging and spectroscopy program. And there we have parts of some of these beamlines. So we have MOUs with the hard X-ray nanoprobe, submicron resolution, X-ray spectroscopy, and the tender energy X-ray absorption spectroscopy, and the full field X-ray imaging XFI. The X-ray fluorescence microprobe XFM, situated at a bending magnet, is totally dedicated to the CBMS program as of recently. I would say that all the structural biology beamlines, they do have Eiger detectors. They are all on insertion devices, except for the X-ray footprinting program, which is on a bending magnet and doesn't have a monochromatic beam. XFP has a pink beam, which they explore for to actually study the radiation damage and how that affects the structure of biological samples. So let's board the train and go through our how to actually get beam time on one of these beamlines. The first stop is submitting a proposal. You can consult the user guide at the NSLS2 webpage. Like Sylvia showed for SSRL, we have preferable 3 types of proposals: a general user proposal, rapid access, and a block allocation proposal, and a proprietary proposal. These are basically the 3 that we have been entertained. On the scattering beamline LIX, we have a collaboration with the Neutron facilities and Oak Ridge, and you can submit a single proposal, both for X-rays and for neutron scattering. So in order for you to submit a proposal, you have to actually enter the PASS system and authenticate yourself, say that you are an actual person, an individual that really wants to do science, by using one of these 3 authentication methods. If you are a new user, you probably will not have a BNL computer account. This in common participant or in common federation is a list of institutions that are registered with our guest, user, and visitor center. So if you belong to one of them, you can use the existence of this way, or you would register with a Gmail account. One of the problems that we have here is that whatever email you use in order to actually submit your proposal, you should really use the email that you will consult frequently. I know it can be an annoyance for you to be inundated by messages from NSLS2 and SSRL, ALS, APS, or CHESS. Everybody telling you something important. However, if that's not done and you do not consult frequently whatever email you provide to us, you may miss the schedule, you may miss then registration in our Gov Center. So it causes a lot of problem. So make sure that you provide an email that you will consult frequently. The other thing that you need to do is to actually save the proposal number that you will get as you submit your proposal, because that is what you are going to share with your team. In order for any member of your group to have access to later on to your to do measurements at NSLS2, they do need to belong to a group, which in this case is exemplified by the proposal. So up to our next stop, which is how to become a actual valid user. But before that, I just want to comment on these proposals. And like Sylvia mentioned, the block allocation groups, which is the type of proposals that we prefer. It's valid for 2 years. It is usually a group of several PIs and can choose up to 5 beamlines. However, for every cycle that we have, you will need to request the beam time. We have 3 cycles. One that where you submit proposals or you submit a beam time request. One that has to be submitted at the end of January, so January 31st, for the summer cycle. Then in spring, May 31st, which is coming up, for the fall cycle, and the fall one, September 30th, for the winter spring cycle. Then you have like a rapid access. It's a project that is, you got crystals, you want to know something about them. This is a project that's valid just for 1 cycle and for a very specific beam time need, and you can only select 1 beam. And then, of course, the general users with proposals that we actually know for many years, which is a proposal that is a single PI. It is valid for 3 cycles and can select. Well, they actually can select, in the case of structural biology, you can select up to 5 beamlines as well. And proprietary use, if you want to work If you don't want to publish and you would like to submit a patent, that is the way to go. Usually companies, they choose these ones, proprietary proposals. So now we arrived on our second stop of our train line. This is where you actually get a guest number or a live number, as it's also known. On this link here, you will have a form that you need to complete and submit. Once you submit that, you get a temporary number that is started with GR and has 6 digits that is used for tracking. And you will get many emails from this address, the govcenter@bnl.gov, which will be requesting some kind of identification, or will, in the end, will tell you your request has been approved. And in that email, you will get several further instructions to actually get to the next step, which will allow you to access all the beamlines that we mentioned before. They can be accessed remotely. The environmental imaging beamlines, they don't really like too much the remote access, but all the structural biology beamlines, they are, I would say, over 95% accessed remotely. So your proposal is still running in the background to be approved. Has been approved. Your request for a BNL guest appointment has been approved. So what now? How do I still access the beamline? So if you only want to access the beamlines remotely, you have to complete these 2 computer trainings online, and then you will be able to request a BNL account. If you are planning, or even if while your proposal is valid and you would like to come to BNL, then you will need to actually complete these other 3 trainings in order to get to the experimental floor. If you miss the email from the Gov Center, you can also access the BNL account through this link, and you can request the BNL domain account, filling out the request doc that you have here. But this should have been sent to you, how to do this, by the Gov Center. Very important is what we are going to see and have to act upon is our safety approval form. Again, you will need to access the PASS system, which is right now the basis for all our proposals and all our interaction with the NSLS2 user office. There you will, so you selected in your proposal, you selected the beamlines. So basically, in order to create this safety approval form, you will go to a tab that's called My SAFs, select the proposal. Maybe you have several different proposals. You will select the beamline. And then if it's structural biology, choose this safety approval form it's called envelope. What this does is it will ask you several questions to which you should answer always. Know if your samples are non-hazardous. And in the end, it will be approved, and it's like very easy. In case if there's any hazard, if you feel that your safety approval, that your sample is not so safe, with some kind of hazard in it, you will need to submit what we call this standard safety approval form. And that has to be submitted at least 14 days before your beam time. So this is something that really needs to be kept in mind. And basically you will know that as soon as your proposal got approved, you can just then select to either submit an envelope or a standard safety approval form. These safety approval forms are valid for a year. However, they need to be submitted for each beamline that you are going to use for your experiment. And our final stop coming up is actually the beamline. So each of the proposals is assigned to a local contact that will assist you in performing your experiment. They will review with you like the sample conditioning, how to ship the samples, how to actually identify your samples through a spreadsheet that we will then use during the data collection. And we'll assign the data to the sample, to the proper sample. It will also, he or she will also review with you how to connect remotely, or in case you are here, how to connect to the workstations at the beamline. We will review the control software and data retrieval. So we have several points of training, one of which is the CBMS Structural Biology Workbench, which we have one coming up now, June 3rd through June 6th. We also have a few training sessions on the beamlines. It's slightly different for starting this cycle. We will be having a dedicated mini shift every month, and you can register for those and actually start manipulating the control software on samples that we provide, or you can also send your own samples for these training sessions. The same valid for the workbench. And finally, we have training videos that you can find on the wiki page that is advertised on the center webpage. Something that's very different from what you saw that Sylvia presented for SSRL, and I believe at other light sources as well, we only accept the spine magnetic bases and unipucks. So the spine magnetic bases are these magnetic bases that have a hollow part in them, and we only accept Unipacks. Then we have some, you can see some of them here. If you send your Unipacks to us, they will be engraved with their names next to their actually name. We are having a two-dimensional sign that can be read then, that's read by the, we have an automated a reader installed at the beamlines so that you can actually be able to have samples from several groups in the liquid nitrogen viewer, and you can then assign the different positions to different users. Very important for your data collection if you are remote is the resolution of your screen. Here's a summary of the beamlines. The ones that are highlighted are our partner users, and you can see the MX ones. The energy range new this year is that AMX has reduced its energy range from, so it's right now from 10.5 to 15 and has a standard beam size of 7 by 5 microns. This beamline is dedicated to automation and high throughput, and will be dedicated to fragment screening. FMX has variable beam size. It can reach between 5 and 18. It is a flexible beamline that can do room temperature and multiple crystallography or serial crystallography. And NYX is a standard X-ray crystallography beamline with 20 by 20 microns beam size. The energy at the moment is fixed at the selenium edge. And you can read more here. I think I will, in the name of time, I can see Sarah impatiently. So we can do this, but I really would like to mention one more thing. It's really the bioimaging, where you can do a lot of X-ray fluorescence imaging, as well as spectroscopy. You can get really fantastic images and see the distribution of different elements in them. This is what we can cover at our center, and QCT is a soft X-ray tomography beamline that's going to come online in the next 3 years. We also have some cryo-EM, and this is a beamline that has very, can do also X-ray I cannot pronounce it, phytography. So anyway, I thank you all for your attention. I thank you for, I thank my group and our user community and the funding agencies. Any questions, you can contact me. I am the contact for all the user program training and outreach for the CBMS group. Sorry for going over, Sarah.
That's okay. I hope I wasn't really threatening.
No.
Oh, thank you so much, Vivian. This is really great. And of course, we do have these recorded. So we're going to post these. And again, I think we're going to, in the interest of time, wait on questions until the end of everybody. So next up on our last speaker is Jenny Wireman, who is the director at Mac Chess at Cornell. And so we'll just wait for Vivian to make sure you're okay to share your slides. Jenny, thank you for being with us. Actually, you know, just in case people do have to leave, I want to say thank you to all of our synchrotron representation who has been here and presented. It's, I think, really helpful for kind of our user community to have a good sense of how to kind of get access and who to ask for help. So thank you. Take it away,
Thanks,
Jenny.
Sarah. Oh, no, you're totally fine. First, I want to just shout out to Vivian and Sylvia, first of all, for both wonderful presentations, and it makes my presentation go very quickly. Second, I know we're very much over time, so what I'm hoping is that I'm going to fly through some of these slides, but I am going to give Sarah and the organizers my slides. There is a lot of links in there, and so I'm hoping that even though I'm going to go very quickly through some of these slides, I'm hoping that you can pick them up and check out some of the fun information that I have in there. So hi, Sarah, thanks for introducing me. I'm Jenny Wireman. I'm director of the Macromolecular Science Facility here at Cornell's High Energy Synchrotron Source. So in case you aren't, okay. There we go. In case you're not familiar with where we are in the US, we're up in the Northeast. We are in upstate New York. We are actually in Ithaca, New York, and we are hosted on Cornell's campus. That's just a picture of the campus. And if you overlay where our accelerator is and our synchrotron, it's actually underneath our athletic fields here. And you can see, I'm gonna need your laser pointer. Here we go. Oh, I've got two. 1 minute. I don't think I've ever had two before. There we go. Okay. So you can see the ring underneath, and our building is actually right here. The rest of this is all Cornell's campus and the beautiful Cayuga Lake in the background. So our facility actually has 3 separate science partners within the facility. CHEX is funded by the NSF. You have the NIH and the NSF funding macromolecular science, and the NIH funds our Mac Chess facility. And then we also have the Air Force Research Lab. We're actually a really small facility compared to many of the other facilities you might have heard of. We only have 8 beamlines currently. So we're really small. And as far as structural biology is concerned, we have 2.5, depending on if people are on our spectroscopy beamline. So I'm going to be talking today, of course, about the macromolecular science, or Mac Chess as it's called, at Chess. Our Chess team at large has over 250 faculty scientists and technical experts and whatnot, and we really rely a lot on our ties to the faculty on campus at Cornell. So all of these wonderful people are scientists and technical staff and leadership, but we're a small enough facility, as I said before, that we actually collaborate a lot with each other. And that's something I think quite unique to our facility. At Mac Chess, and here's the team on the left there, we do 2 major categories of structural biology. We do small angle X-ray scattering, and that's biological. This can give you information on molecular shape, the degree of protein folding, protein interactions, and other higher order structures and mesophases, and macromolecular crystallography, which of course you've heard about all day today, which gives you electron density and atomic resolution, including solvents and ligands, and B factors and diffuse scattering. So our website is down here. It will be mentioned a couple more times throughout the presentation, and it is an active link. So when you get this, it'll take you directly to our website. This is our team. We have our kind of core staff here. So our staff scientists and our technical staff, we have a few graduate students that work with us. We have a few visiting scientists, and technically these are visiting scientists who have retired and just can't stay away, which we really enjoy. And we have a wonderful technical staff also that helps from Chess's side. But we have a lot more people who help us out. I won't go into that. So there's a lot of different capabilities at MacChess, and I won't go into these in too much depth because they're all really unique, and I think each one requires a little bit more time to talk about. So with small angle X-ray scattering and biological, again, we get this kind of envelope. We can do SACs and time resolved SACs. We can do size exclusion in line, sex mouse. We do high pressure small angle scattering. So this can actually tell you a lot of information about the folding of your protein. And then we can also do anaerobic SACs. And with anaerobic SACs, we can actually do size exclusion chromatography in line with your anaerobic setup. And this is if you have any kind of proteins which are sensitive to oxygen, you can use our system and do it without any oxygen present. On our macromolecular crystallography beamline, we have kind of the standard remote high throughput crystallography in which we use a band 2 or an ALS style robot. We can do multi-temperature crystallography. We can do serial crystallography at ambient temperature. We can do high pressure cryocooling and high pressure crystallography using a diamond anvil cell. You'll see it here on the right. That's a diamond anvil cell. And we can go up to some pretty high temperature or pressures. And that can actually give you a lot of information on the conformational landscape of different proteins. We've seen different ligands binding with different affinities at higher pressures. We've seen different conformations have different proportions at different pressures. And then we also do diffuse scattering, and this gives you an idea of correlated motions within the protein crystal itself. So this can give you information on dynamics as well, and B factors. Okay, and through all of these techniques, which there are quite a few, we do offer hands-on one-on-one training on all of these techniques. So we absolutely love doing workshops and hosting different techniques. So if you have any question on any of these, especially the more unique, obscure ones, diffuse scattering and high pressure, we're happy to train. We also love to host new techniques and methods. So if you have a system that requires something completely out of the ordinary, we will absolutely work with you to get that kind of a method or technique implemented at either one of our beamlines, whether it's SACs or it's crystallography. We like to think that it's a developer's playground. Some technical specs, I will not go into detail on this slide because you'll be able to look all of this up. But specs on our different energies and sizes of beam, as well as detectors, are here. And the different setups for both of the beamlines are represented down there. All right, so say you want to come to MacChess and be a user and use some of those wonderful capabilities that I've just gone through. First place to stop is our website. It is www.chess.cornell.edu, and you'll pop up to a page that looks like this. There will be several tabbies here at the top. The ones that you're probably interested in is the users and the facilities. So what that looks like then is users are here, and then you'll have access to the user guide. I've also given the link up here. So the user guide has lots of information about becoming a user, about questions, facts, any kind of beamline specifics. So that'll be all within the user guide and links to basically everywhere else on our webpage. If you want to know more specifically about facilities like MacChess, then you just go to the facilities tab and pop on down to the MacChess, and that'll take you to the specifics on the macromolecular program here at Chess. I try to put this a couple of places. If you have any questions, our Chess user office is open most business hours, and they'll happily answer any questions. You can also email me. I'll have my email at the very end of this presentation. Okay, I know this is a lot of words, but basically we have a proposal system, and like Vivian said, every proposal system is different at every facility. So if you go to users on our webpage, you'll see this thing called BeamPass. BeamPass is our version of our proposal system. You can access it from our website, which has our beamline and contact info and beamline schedules. But this is how we get our proposals through. And it's how all of Chess gets their proposals through. They're reviewed for scientific merit and safety based on a peer review system in our safety committee. And the proposal length itself is 2 years or 6 runs. After you've submitted a proposal, and I'll show you how to do that in a minute, then you can submit beam time requests. And that's how you actually get allocated time at our beam lines. And so this you can do prior to the run. You can select as many shifts as you want. They're 8-hour shifts each. And then you can update samples that you might have proposed in your proposal. However, MacChess is really special because we have this thing called rapid access. And this is where you can submit a proposal and a beam time request basically anytime. So in the middle of the run, like right now, you can submit these. And it should, when you put these in, your proposal title should start with rapid access. And I'll show you what that looks like in a second. But yeah, so this is for samples that can be available on short notice, or say, you weren't getting crystals, you suddenly get crystals and they don't last long and you want to, or their quality doesn't last long and you want to send them in. This would be the way to do it. Okay, so you want to get started in the proposal system. Oh, I forgot, sorry. One more thing is some interesting dates. I'm not going to go through this again because you can look at this, but our next run starts in September. And the call for proposals and beam time requests will begin in a couple of weeks, and that'll be open until July 17th. Okay, but again, rapid access or proposals are selectively accepted at any time. I can answer questions about that. All right, so the first thing you do if you want to come to Chess is you need to create a user account, and we call it a class ID for historical reasons, but go with me on this. So you'll need your own ID. So once you go into BeamPass, it's going to ask you this question to log in, but you don't have a class ID. So you'll select this one down here that says don't have a class ID, and it'll pop you into this form. You'll fill out this form and you'll get an email, and the email will have tons of information on how your user account gets set up and how to get into BeamPass. Once you're in BeamPass, you can now create a proposal. So this is the homepage of BeamPass. You'll notice a bunch of options on the left. You'll see active proposals, which there is one, and it is a sample proposal, and then beam time requests and schedule beam time. So there's lots of information right here on the home screen of our BeamPass system. So you want to create a proposal. It's just a click of a button right here. You'll go ahead and click that button, and it'll pop this up. And so there's various fields you'll be asked to fill out, including basics like the proposal title. Now remember, if this is a rapid access, you can put rapid access in here, and it'll be shuttled through a rapid access system. And if it's not rapid access, just go ahead and put the proposal title. Usually it's nice to have some sort of science clip in there that helps describe what the project is. But then there's several other data fields, and you'll fill all of those out as well. The more detailed you are, the better, specifically in the scientific justification and in the experimental plan. And as somebody who's on the safety committee, I love it when people are very specific about what their specimens are and if they have any hazards in them. So, and once you submit that, it goes to our user office, which then eventually sends it to peer review. Once it's peer reviewed and our safety committee looks over it, then you're able to create a beam time request. So creating a beam time request looks like this. You'll be in your home again, and there is a button that will say create beam time request. I think I actually showed it here. There it is. Start a new beam time request, and then it'll have this drop down and it'll show you an eligible proposal. So this just happens to be one of my proposals that I was a part of. And then it shows you a create a beam time request, or BTR. So you'll click that button and what pops up is this screen. And then you'll go ahead and select the run cycle that you want to have it in, the requested dates. This is rapid access for right now. So that would be a rapid access proposal and a rapid access beam time request. You'll put in the dates that you want, estimated hours, and then whether or not it's mail-in or joint, and if you're going to be in person or remote. Pretty easy questions, I think. The rest of this is all questions you probably would have filled out in your proposal. So you can just copy paste. That's pretty easy. All right, that's it. Once you submit the beam time request, it goes to our scheduling people, and then you'll get emails and whatnot about when your beam time is, and when your beam time is, and how to get lots of details. So we usually inundate you with too much information. All right, so for macromolecular sample preparation, I'm going to focus on that because I know there's not a whole lot of SACS people here, but I do see somebody talking about SACS. Ooh, that's Sarah. Okay, I'm going to focus on macromolecular crystallography. We tell you about what pin height, that is mostly so that you can center your crystal better in our crystal centering program. We actually accept quite a large range of different pin bases, basically almost everything except for we do not offer mini spine compatibility, but that's, I guess, not too unusual. Samples can be shipped in ALS, in Uni, or MSc pucks. You'll see them down here. This is our Band 2 automounters. So this is our robot, which gives us the capability of being fully remote for our users. And you'll see that it holds 9 pucks and it can accept these MSc pucks. We do have kits. So we have kits that we can loan to users who don't have their own pucks and tools. If you need a loaner kit, be sure to request it in advance because sometimes it takes a long time to ship, as Sylvia told you. And also give you time to familiarize yourself with tools and load your samples. We also do non-standard crystallography sample preparation. A lot of this is in conjunction with users who bring in interesting things like new mixing jets or chips or capillaries or plates. But we'll work with you, especially if you're in person, to be able to actually get these things, these different devices mounted on our beam lines. We have a lot of flexibility on our beam lines to kind of tear stuff apart and put your samples in. We also do room temperature serial crystallography. We love the compatible goniometer bases because that means we don't have to tear things apart in order to put your samples on. But of course, we're also happy to do kind of more development and kind of cutting edge research as well. So, okay. For shipping samples, half of our users do these cryo-cooled samples. A little over half of our do cryo-cooled samples, and they'll ship them indoors, and that's kind of the standard. Sylvia shared with you these room temperature sample holders, and we'll support that for shipping your samples to the facility. But almost all of our room temperature sample users actually come and collect data here themselves. So we don't have remote capability for room temperature, but we do have a humidity chamber as well as capable of accepting these shipments. In some special cases, we've collected data for users, but those are kind of special cases and not a lot of crystals. So, but just to give you the idea, we are moving in this direction of having better room temperature, humidity controlled sample environment at our beamline. Okay, we do have a shipping form, and I've linked to it here, so you can click that when you get to the point of shipping things, and it covers all of these different issues. You'll print them out. I think Sylvia already touched on it. We only receive shipments Monday through Friday, so please don't ship it so it arrives on the weekend. And she already showed you how to do all the dry doers that are charged well. So we recommend that you don't use the US Postal Service. We've had so many different awful situations with it, but we typically have our users use FedEx. We've also had DHL to some really good results. So, okay. For data collection and processing, it looks a lot like many other types of data processing. We have a crystal click to center, we have an automounter screen, we have diffraction viewers, and we have capabilities like raster scanning where you can take and look at your mount and see if there's any crystalline diffracting areas. So I'm not going to go too much into this since I don't have time. But we do have some fully automated data processing using ZIA 2, but we have a suite of different processing that you can also use in our servers. as well as available to you to use from your remote session. We do data transfer via Globus, but we also use rsync. Those links are here and we have instructions. Again, the links are live. So when you get this presentation, feel free to click through to see more of the complete instructions for both of these. We do remote access. Like many, we use No Machine. However, we have a dual device manager. This is to provide security for our No Machine access. And this is just using that class ID that I mentioned at the very beginning. It's the same thing that you use for BeamPass, so it's not anything different, but you have to have Duo enabled on your phone. And so that you can accept and approve the access to our No Machine. Again, there's lots of instructions here, and I don't want to go through them because, well, they're boring. But they are instructions that are necessary to get into No Machine and to get into our host. So please read these if you're wanting to access our Beamlines. I wanted to highlight again the capabilities just as a parting message of we have a lot of really fun stuff going on, a lot of kind of unique capabilities for doing really fun science. We love extreme stuff. We love doing high temperature. We love doing high pressure. We love different pHs. Yeah, you name it. We like to have fun. We work in collaboration with the NSF funded program called CHECKS, and they fund and support an extreme biology beamline. So we're a part of that collaboration. So again, hands-on technique. CHESS is supported by the NSF award here. That's the CHECKS award. And then MacChess is supported by the NIH award here. I want to thank you guys all for whipping through that set of slides. I don't want to keep us too much longer. I don't think that I stuck too far outside of our time or my time. Yeah, and if you have any questions, I'm happy to take them.
Thank you, Jenny. That's wonderful.
Phew.
I want to open up the floor for any potential questions from anybody. So I guess one question that I have, and maybe on behalf of our users, is how difficult is it to get time? Is it competitive? Do you have to wait forever for both like chess and NSLS too? Because Sylvia had to leave, so I'll just ask you guys.
I can go first. So it's gotten a little bit more hectic with the APS being down. We've had our oversubscriptions have gone a little bit haywire. However, APS should be coming back online this fall. So we do, yeah. But the thing is, we kind of keep a waiting list of groups which, who've been approved and, they've gone through the system. And groups will cancel their beam times. We have several cancellations typically in every run just because people will not have crystal samples or, someone gets COVID and I don't know, someone breaks their arm. Someone broke their arm once and couldn't come to their beam time. So they'll cancel. And in that case, that's when rapid proposals can be, extremely useful. We go through the waiting list. We keep a waiting list that's kind of ongoing because sometimes people can't make a specific beam time. And so we'll just kind of keep the wait list and then go down the wait list until we find someone who can fill in that time.
Great. That's great.
I would second Jenny. That's basically also what we have been observing. When we have a cancellation, we try to schedule people who are on the waiting list.
I think I just wanted to make sure that, our users heard that, that they shouldn't be intimidated by, oh my gosh, it's going to be so competitive. I don't know how to get time. That the, beam lines and the facilities and Crystallization Center and all of these facilities, we all really want to help people do their science. And so, so reach out to us with questions, reach out to the beamline scientists and to Jenny and Vivian and Sylvia and Ina to try to kind of get help if you need it. So.
Well, I have a question actually, because I think maybe Jenny can. Are you going to implement the universal proposal system in chess? I know
I mean,
APS is using it.
we don't have plans for it now because it's currently it would need to be a chess-wide implementation. And so, you know, we might be a small facility, but that actually has a lot of that's a lot of change for the larger. We would like to. So if I can, I will push that. So yeah.
All right. You know, I think that we have had a really great day. I really appreciate everybody who has been here and asking all kinds of questions and has been giving great presentations. And if you have additional questions, please do reach out to us. There have been lots of emails. We will probably do a survey about this virtual workshop. Please also answer that survey. That would be really helpful for us so that we can plan additional workshops. And at this point, I think, you know, I put a couple of links in during the last little bit about, we do have some on-site workshops coming up as well that are follow-up on this. The June workshop is already full, but there are still some spots in August, and so the link is in the chat, or you can go to our website, getacrystal.org, if you're interested in applying for one of those spots for the August workshop. And again, I really want to say thank you to our speakers, say thank you to NIH for funding our national resource, and to say thank you to our sponsors, Hampton Research, Formulatrix, SPT LabTech, and Mitogen. We are really very grateful for the support, for being able to kind of do these types of engagements and outreach. And so with that, if there's no further questions, we will kind of close things up. Thank you so much for attending for our workshop.
Thanks,
Thank
Sarah.
you.
Talk to you later. Bye-bye.
Thank you, Chuck.
Thank you
Nice
all.
to see you.
Bye-bye.
Bye, everyone. Thank you so much.
All right. Well, I think I'm going to end the meeting. So thank you, everybody.
Thank you, Sarah, for hosting, well, for all of you for hosting such a wonderful workshop so far.
We're excited. It's I think our first one ever. So on site, you know.
Yeah. All right.
All right.
Ciao.
Talk to you soon.